Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Results? You've seen the incomplete picture of short reads. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture.
Right. Very good morning to all participants. Welcome to the second days of the 4th International Conference on Molecular Biology and Biotechnology, in conjunction with the 27th Scientific Meeting of the Malaysian Society for Molecular Biology and Biotechnology. First of all, I would like to introduce myself. My name is Boon Jin from the University of Malaya, and I will be the MC today. On behalf of the organizers, I would like to extend my warm welcome to all the participants to the 4th ICMBB 2021. Before we start our conference, I would like to remind all participants of the house rules. Please mute your videos and audio. You will be given instructions if you need to turn on your video. If you leave the room and want to get back in, or you want to move to the other room, you have to click on the link in the program and you can move in. If you have questions to ask during Q&A sessions, please type your questions in the chat box and to whom it is addressed. Ladies and gentlemen, to begin, we have two MSMBB EduSTEM grants winners to share their projects on STEM education with us. For your information, the MSMBB EduSTEM grants was set up to support projects working with science, technology, engineering, and mathematics educations, and to fund activities which are aligned with the United Nations Sustainable Development Goals. You will find more information about these EduSTEM grants in the closing ceremony. Without further ado, let's welcome our first winner of the MSMBB EduSTEM grant, Associate Professor Dr. Siti Sarah Othman. The title of her project is, Are You Human or AI? See Your DNA.
Assalamualaikum and a very good morning, everyone. I am Sarah Othman from the Department of Cell and Molecular Biology, Faculty of Biotechnology and Biomolecular Sciences, University Putra, Malaysia. I'd like to take this opportunity to extend my heartfelt gratitude to the organizer and to the Malaysian Society for Molecular Biology and Biotechnology, MSMBB, for allowing us to share our journey through the MSMBB EduSTEM Grant 2019 entitled, Are You Human or AI? See Your DNA. This project is a development of a science educational prototype, a homemade DNA extraction kit to increase science literacy in the community. So, what is science literacy? Science literacy is a crucial skill to be acquired by community members to recognize obstacles and to summarize conclusions based on evidence in order to assist them in making everyday decisions towards environmental changes caused by human activities. In general, empowerment of science literacy will allow each individual to provide expedient resolutions towards life and their ecosystem that can be beneficial to all. Parallel with this imperative need, science curriculum has been embedded in the educational system as young as from the preschool to the university, with the curriculum being highly specific and niche-oriented. However, it was observed that traditional teaching in the school curriculum and the lack of interest of the current community in science generally fail to garner more individuals to be interested in science, and this resulted in poor science awareness, read literacy, among the public. So, what should we do? How do we play our part? In order to engage and encourage community members, from parents to children, to enhance their interest in science literacy, a simple, easy, and economical experiments relevant to their daily basis can be developed. In this project, our team is thinking to bring them to know themselves further, to be able to see their own DNA, the genetic code of life. At the start of this project, We develop a homemade DNA extraction kit and test them within the community, starting with primary and secondary schools around Klang Valley and several sciences fair organized by the local community. The kit will be developed by two universities, University Putra Malaysia and University Sains Islam Malaysia, and the exhibitions will be conducted by us, the researchers, MSMBB members, and also the university students. Aligned with the nation interest in increasing STEM awareness in STEM education, increasing awareness in STEM education, this kit will be developed as an educational product that will be benefit in fostering creativity and allowing community members to be innovative and make science relevant to their daily life. The project has two objectives, which are to develop simple, easy, safe, and economical homemade extraction kit, and to foster creativity and engagement within community members through science activities that is related to their daily life. Let me share some of our pictures. I hope everyone can see my slide. So in 2019, at the start of this project, together with my team members, Associate Professor Dr. Chia Swet Lin, known as Dr. Eddie from the University Putra Malaysia, and Dr. Muhammad Zami Othman, known as Dr. Zami from University Sains Islam Malaysia, we have visited several local and international schools, and we have also managed to join several public engagements to demonstrate our kit. For the first phase, around 100 secondary students of Maktab Rendah Sains Mara Serting in Negeri Sembilan, and another 100 of students from the age of 6 to 9 years old from the Nilai International School have experienced the STEM activities ranging from DNA purification, imaging, and also fun puzzle to increase their understanding on the genetic code of life. So as you can see the image. And we have also managed to join the Fast Lab UC during the Kuala Lumpur Engineering and Science Fair in 2019 at the MIECC Selangor. In this science fair, we had managed to train at least 10 undergraduate students to facilitate our kit and receive more than 1,000 visitors throughout the exhibition, regardless whether they are students, their parents, children as young as 5 to 6 years old. It was really, really interesting and engaging fairs at that stage. For the second phase of the project, with an objective to bridge borderless science through STEM education, our team has been given a chance to collaborate again with the Fast Lab UC and Dr. Terasa Ekobon from the Department of Genetics, Faculty of Sciences, Kasasat University, Bangkok, Thailand. We have managed to expose and train the staff and the postgraduate students of the department on how to conduct STEM workshop for the school students around Bangkok and also to the public. During our visit to Bangkok in February 2020, just before the lockdown, we have been invited by Tanyara School at Patung Thani to participate in their STEM Open Day. It was a golden opportunity for us to demonstrate STEM activities regionally. The team has also managed to train local postgraduate students of Kasasat University to facilitate in that event and also to other schools in Bangkok. We are proud to say that throughout our visit, we have been waving the MSMBB flag with us. As you can see, we'll be bringing the banners of MSMBB flag. For the final phase of the project, which is to further refine the kit, we have included and trained an undergraduate student to incorporate this as a final year project for her to complete her degree. Ms. Qurratu Ain Shafizam has managed to develop a prototype kit, organize several public engagement exhibitions, including in Minggu Sains Negara this year, and workshops, and also has also completed market surveys by posting the kit and recording comments on this kit from relevant stakeholders. At the end of the project, we are currently working for the improvement of the kit packaging, and also necessary intellectual property application towards the final commercially available version of the kit. We have also shared this kit through several social media platforms. So in these slides, Ms. Kurato Ain have joined the MSMBB TikTok competition 2020, and her video has been posted in the MSMBB official page. And we also have one posting from TikTok, and I would like to share this video with you today. Let's just watch this video for a bit.
Is an experiment called Are You a Human or an AI? We have shot a car, a biscuit, before silicate, some pineapple juice, and some little yeah. Yes. Yeah. Okay. Let's do the axle.
What is this?
Okay, it's a DNA.
This is your DNA. So that proves you are you human or AI?
Human.
Human. We can see this DNA because DNA cannot be diluted in alcohol. That's why it's there.
I hope that was clear and fun. Thank you, Luz Ryans, the model for this video. Well, with that, we would like to express our gratitude again to the Malaysian Society for Molecular Biology and Biotechnology for giving us a chance to explore the possibilities for our team to achieve in promoting STEM education to the public. The EduSTEM grant has tremendously helped us to elevate our interests and project to another level. And we hope to create more opportunities in the future to train and create awareness in this field. I would also like to thank my team members, Dr. Eddie and Dr. Zami, together with the FAST Lab, Yusim, Ms. Nazura, Ms. Sharifa from UPM, Ms. Farahani, and Ms. Kuratun for their contribution to this project. If you have any questions, inquiries, or if you'd like to join our project, feel free to email me at sarahouthman@upm.edu.my. So with that, I would like to say thank you for your time listening to me. Take care, stay safe, and goodbye.
Right, thank you, Dr. Siti Sarah Hoffman, for sharing her EduSTEM projects. Our second winner is Dr. Adeline Song Ailian. She's going to share with us her EduSTEM projects with the title, Evoking an Interest in STEM Through the Mysterious World of the Unseen. Please welcome Dr. Adeline.
Hi everyone. Today I would like to share with you a project that we ran under the MSMBB EduSTEM grant, and the title of our project was Evoking an Interest in STEM Through the Mysterious World of the Unseen. So before I begin, I would like to introduce the team members on this project. I'm Adeline, and together with me I have Dr. Suryana Sabri and Dr. Saira Ismail, and we are from the Department of Microbiology, the Faculty of Biotechnology and Biomolecular Sciences in UPM. So the project which was initially proposed was to conduct three workshops, microbiology STEM workshops in 3 different rural schools. And the main activity which was proposed was to conduct a full scope workshop. So for those of you who are not familiar with full scope is a paper microscope, which is very affordable because it's only around RM20 in Malaysia. And this microscope can be folded like origami, and once it is folded, it's a functional microscope which can magnify up to about 140 times. So it is a very useful microscope for students and for anyone interested in science to actually look at samples around them, as they can prepare the slides at home and view it under this paper microscope. So together with this full scope microscope, this full scope workshop, we also proposed to run a micrograph competition because this full scope can actually be attached to your mobile phone, where you can take pictures and even videos of your specimen that you are viewing under the full scope. So other than this micrograph competition, we also plan to run a series of different STEM activities with a budget of 5,000 ringgit. So this was the first school that we visited in SK Bukit Rahmat, Tongkang Pecah in Johor. And this was conducted sometime in February 2020, just before the first MCO in Malaysia. So we were able to physically go to school and run the workshop with these students, about 100 primary school students. So apart from the people from UPM going to the school, we also invited industries to join us. As for example, Full Scope Malaysia, once we pitched the idea to them, they actually sent some of their members to also join us in Johor to conduct this workshop together. And Full Scope Malaysia, we actually bought a few some full scopes from them, but they also donated more full scopes so that we could engage more students. And then we also had Yakult Malaysia, who came and gave a talk on probiotics to the students, and also to treat the students with Yakult. So this was a team that we brought down from UPM, a group of 3 lecturers and 8 postgraduate students. And this was the first workshop that we conducted. Unfortunately, after this particular workshop, the MCO happened, so we had to switch our strategy of conducting this workshop. So what we did was we incorporated the workshop the project for this grant into a course that we have in UPM called Service Learning in Microbiology. So this is a SULAM course. SULAM stands for Service Learning Malaysia, University for Society, and it is a compulsory course which is incorporated into the curriculum of the program of the Bachelor of Science Microbiology with Honours. It is a 1-credit program, and this is the program that we are actually teaching. So in this course, the students are supposed to translate whatever microbiology knowledge they have gained over the years to community engagement. So at this point of time, our undergraduate students were all over the country, back in their homes mostly. And so we gave them the flexibility to actually go and find their own community of school children that they want to engage, and to run STEM workshops with them with a focus in microbiology. So we told them that the compulsory activity to run is the full scope workshop, but other than that, they can also run any other activities that they want. So these are some of the examples that were chosen by the students, and some of the students, depending on their situation, they actually did it face-to-face, and some of them did it online, because some of them were like maybe stuck in the university hostels. So these are some of the examples of activities done. They isolated bacteria, so we actually send them the agar powder by mail together with the full scope, and then they prepared all this in their their kitchen. So although they didn't have like an autoclave or anything, they were able to just use, just heat up on normal stove and to prepare all this media, and they were able to actually grow bacteria to show the school students. And these are other examples of the activities done. This is a Vinogradsky column, which is to show different diverse bacteria growing on different carbon sources. Then they grew different types of, they prepared food with different microbe elements in them. For example, mushroom and tempeh. They managed to isolate even luminescence bacteria from squid, and so on and so forth. So for both times that we actually did the activities, we also run this micrograph competition. So the students were asked to submit their micrographs, which they had taken from their full scope, either pictures or videos like this. and then submit it to us, and we choose a winner out of the submissions. And this is just to encourage the students to continue to use their full scopes even after the workshop has ended. So the output of the project so far, we've reached about 170 school students. And of course, other than inculcating interest in STEM and especially in microbiology in the school students, we also manage, I think our microbiology undergraduate students who actually did this project for their service learning, they actually had very good feedback on the program because they attained a lot of skills such as problem solving skills. They had to communicate science to children who had no background in science. They had to plan their own protocols and optimize their own experiments. So the students gave very good feedback because they said that this was some students even said that they realized that after doing this program, studying is not just about scoring A's in their exams, but also to share knowledge, especially with the community. So other than that, of course, as I mentioned earlier, we had engagement with the industry and also with the community, and the students, because some of our undergraduate students, they had very good Sulam projects, so they actually used the projects to enter into this national competition called Sulam Innovate. So I just would like to encourage everyone to actually apply for this MSMBB grant. And we would really like to thank MSMBB for this opportunity, for giving us the opportunity and also the funding to run this kind of program. So with that, thank you very much.
Right. Thank you, Dr. Adeline Song, for her interesting presentations. Before we move on to our keynote speaker, may I request you to switch on your camera for a while, if you don't mind, to take a group photo. Give us a few seconds. Our technical team will screenshot.
Hey, how to do the love sign thing? Ada? Macam ni? Okay. Tahu.
Okay, it will take a while because there are a lot of screen here. Okay, right, done. Thank you very much. Next, I would like to invite our keynote speaker, Professor Dr. Georges, to share with us his talk on the trumps and vicissitudes of biotechnologies on malaria control. Prof. Dr. Georges' scientific career has been dedicated to the studies of Plasmodium protozoan parasites, the causative agents of malaria. His interest in these pathogens does not only rest on their major impact on public health, but also because they offer a quintessential models for fundamental biological studies. He actively pursues investigations that address 4 broad topics, including the genetic diversity of Plasmodium species that infect humans, the biologies and immunologies of the hepatic stage of the infections, the pathogenesis of cerebral membrane, malaria, and the molecular characterizations of Plasmodium species in higher primates. Prof. Georges has published more than 150 research papers and has a H-index of 57. Ladies and gentlemen, please join me to welcome Prof. Georges. Prof. Georges, you have 30 minutes and you will hear a bell 5 minutes before the times. There will be no Q&A sessions for keynote speaker. Thank you.
Right. I'm just trying to, good morning. I would like you to confirm that the screen is visible
Yes.
to you.
Yeah, we can see your screen. Yeah,
excellent. Allow me to go. Well, good morning, and thank you very much for inviting me to present some of my thoughts to you on the biotechnologies and malaria control. And I have titled this as Triumphs and Vicissitude because with respect to malaria, there has been some good things that biotechnology given us, and it kind of put some spokes in our wheels at some stages. So let me start by defining what biotechnology is and how I consider it. It is, as you can read here, it's an applied science using living organism that people have used to obtain either a product or a process. In the case of malaria, and for scientific reasons, it is also a way to acquire knowledge. And in malaria, this has certainly been the case. Now, I can see from the abstract book that, excuse me a second, that many of you are not malaria experts or malariologists. So I would like to start with a brief introduction of malaria and how to control it. And it will be brief, and then talk about the emergence of modern biotechnology and its effect on the malaria community, and then briefly discuss what is happening currently. So first of all, malaria is, for those of you who don't know, is a blood disease caused by a protozoan. It's basically a parasite, a protozoan parasites that infects red blood cells, grows into them, and then they burst, and then this is what gives us the disease. This was known since antiquity, but only discovered in 1880 by a French medical doctor who was working at the time in Algeria. Since within the years after he discovered it, it was discovered that there were 4 different species of Plasmodium parasites that infect humans. It took about 20 years before two scientists independently, one Ronald Ross working in India and Grassi working in Italy, discovered that it was transmitted from man to man via the bite of a mosquito. And it was discovered also that the only mosquitoes who could transmit it are those that are of the Anopheles genus. Today, we know that there are more than 50 species that are major vectors of malaria. And of course, there are many more that are minor vectors of malaria. So it is quite it has quite a diversity of ways of it to be transmitted. Now, the life cycle of malaria has taken many years to uncover. After the discovery in the late 1890s of the transmission stages, it took another 50 years before they discovered what happened to the parasite once the mosquito bites the person and inoculates it in it. So only in 1948, they discovered that the parasite goes to the liver as the first initial step of its cycle. And it took another 50 years also to discover that in some species of parasites, these liver stages can stay dormant for many, many years. And this is the reason why many people think that malaria lasts for a lifetime. Now, this is what the life cycle of the malaria parasite is as we know it today. As you can see, we know much of the details of what happens with this parasite. I don't know whether you can see the pointer, but I mean, these are the blood stages that we see in humans that give the disease. And this is how they transmit via the mosquitoes. And the steps are, as you can see, quite diverse, relatively complex, and the parasite adopts many different forms throughout its life cycle. But for the purposes of controlling malaria, one can consider malaria as basically a tale of 2 bites. If a carrier who has the parasites in his blood is bitten by a mosquito, that mosquito is likely to pick up sexual stages of the parasite, which within 12 to 13 days will grow to form the infective parasites that go to the salivary glands. And when that mosquito bites another person, that person becomes a new carrier and gets the disease. So how do we control malaria? Well, it's a very simple process to control malaria. You either attack the parasite, or attack the mosquitoes, or do both. And up till the beginning of last century, quinine, which had been discovered in the 17th century, was the only true effective drug against malaria. And with respect to, once we knew that the mosquito was the one that transmitted it, people had drained the swamps or the areas around them, or they screened their houses to prevent malaria from the mosquitoes from biting them. As you can see, these two pictures are taken from a publicity campaign in the First World War, sorry, because during that time, it became obvious to a lot of to the people who were trying to control malaria that quinine was really not the most, although it was an effective drug, it wasn't the best drug to take. So the first contributions that biotechnology made to malaria was the chemical biotechnology. The Germans were at the time the best chemists in the world. The German chemistry industry was extremely advanced compared to all the other European and nations in the world. And after the Second World War, they embarked on a large project in which they wanted to make synthetic malarias. And in 1926, they successfully made plasmocin, and then followed that by atebrin. And these two drugs have been used for a while, but they were not very good. And by the 1930s and 40s, chloroquine was discovered and made and tested during the Second World War, and found to be a highly effective, cheap drug that one use against malaria. Towards the end of the 1930s in Switzerland, Paul Müller has discovered also a compound we know as DDT, which proved to be an extremely effective insecticide. It can be sprayed and it will kill practically all insects very quickly, and it is also very cheap to make. The fact that these two compounds have been discovered has spurred has led to people considering that now, with an effective anti-malarials and with an effective anti-mosquito compound, it is possible to eradicate malaria. One has to say that the chemical biotechnology is still giving us today all the drugs that we use against malaria and the new insecticides. So the WHO in the 1950s devised a malaria eradication program, which started in 1955 and which was based on three simple strategies. You diagnose and treat everybody who you can get who has malaria, and at the same time, you spray the area in order to kill the mosquitoes. And they considered that with these two drugs, with chloroquine on one side and with DDT on the other side, they will be able to eliminate malaria within 6 years. It was a success to a large extent, because malaria, which was prevalent throughout the world, at the time that they started this, malaria occurred in nearly all of Europe, including Russia and including the United Kingdom, France. It was very prevalent in Italy, in Greece, and all around the Mediterranean. And over the years, by the early 1960s, malaria had disappeared from nearly all the temperate areas, has decreased substantially in Latin America, decreased to a large extent in Southeast Asia, and wasn't very successfully decreased in Africa. The problem was that the scientists and the WHO had underestimated the adaptability of the parasites on the mosquitoes, who promptly developed resistance, and they failed to appreciate also the difficulties of implementing that sort of program globally. So although it was a kind of a failure of the chemical biotechnology, it nonetheless did eradicate malaria from a large parts of the world. It unfortunately also eradicated malariologists to a certain extent, because people, having considered that malaria is now going to be controlled and eradicated, stopped working on malaria, and the number of malariologists and medical entomologists dropped substantially during the 1960s. After the failure, the recognition that malaria control had failed, the the Malayologists who were there and people who wanted to study the parasites faced two major problems. The first problem was that they had now limited access to parasites. One has to remember that previously a lot of the malaria work was being done by scientists, European scientists who were working in colonial possessions. And therefore they had access to field studies, they had access to a lot of parasite material. With the decolonization and with the fact that malaria therapy had ceased, they could only rely on volunteers or on field studies, which were not that easy to make, and they couldn't get a lot of parasite material. Animal models existed, but they had their own problems. The other limitations they had were technological. If you go back to the 1960s and the early 1970s, the only technologies that malayologists had to study was serology, but just was very simple serology, was microscopy, and protein purification was in its infancy, and in any case, it requires a lot of parasite material. Now, in the 1970s, coincidentally, and luckily for the malaria community, there were four major discoveries that made it easy for them to embark on a new phase of studying malaria. First of all, genetic engineering was initiated basically in the 1970s. In 1970, Hamilton Smith, working in the United States, discovered restriction enzymes. These were the enzymes that help bacteria fight phage invasions, a process that was discovered in 1969 by Salvatore Luria. These restriction enzymes cut DNA in a very specific place and always the same place with the same sequence. Today, we know there are about 200 to 300 different types of restriction enzymes, each cutting DNA in a different place. The fact that you can cut DNA in a specific place meant that you can actually isolate pieces of fragment of DNA, rejoin them together in the order you want, and this is what genetic engineering is. Isolating a piece of DNA from one organism or one gene, and then splicing it into another one. This technology was developed in the 1970s. The second major advance was the discovery of monoclonal antibodies in 1975 by Cesar Milstein and George Curler. What they basically did is that they managed to get a single B cell, which makes antibodies with a single specificity, and expand that by making a hybridoma and growing this up, and then they could purify large quantities of antibodies that are highly specific, hence they're called monoclonal. This now allowed people to tag a protein or to detect a protein or a particular shape in a protein with high specificity and with great ease. The final pan-biological discovery that was made was DNA sequencing. Sequencing of DNA was nearly impossible beforehand. Before the 1970s, the largest piece of DNA that was sequenced was 25 base pairs of one of the bacteriophages, and it took them 3 years to get the sequence of these 24 base pairs. Walter Gilbert in the mid-70s discovered a chemical way to sequence DNA, and then this was improved and in a slightly different way by Frederick Sanger in the United Kingdom, who discovered what is known as the dideoxy method for DNA sequencing. And within a few years, the amount of DNA that one could sequence increased very rapidly, and it became, by the mid-1980s, it became routine. Now, having now these tools without parasites would not have been very useful to the malaria community, and the malaria community needed good quantities of parasites to be able to apply these technologies to their things. This came by the development of in vitro cultivations of parasite by William Traeger and James Jensen, and they've succeeded in 1976 for the first time to grow in vitro, cultivate routinely in vitro a parasite of malaria. And it so happened that that parasite was Plasmodium falciparum, the parasite that infects the human the most and is the most dangerous and is the most widespread of the 4 species that we have. Nowadays, we can cultivate a few more species, but not that many more. It should be said that William Traeger started trying to cultivate parasites in 1943. So it took a long time And it was lucky that it occurred at the same time as these major advances in biotechnology were made. With all these confluence of these advances, it became simple, in a sense, for people, scientists at the time, to rethink the way to control malaria and to move towards the idea that now one should be able to vaccinate against malaria, because the availability of these biotechnologies gave them a strategy to obtain a malaria vaccine. Basically, what the idea was is that you take the genome of the parasite, you put it in an expression library, and then you screen that library with immune serum. You take antibodies from people who are hyperimmunized by malaria because they live in malaria area. The genes that react, the clones that react in the library with the serum will then be sequenced. Therefore, you identify the gene, then you make a recombinant protein, and then you can basically use that as a basis for a vaccine. Now, over the 1980s and the 1990s, this has dominated the malaria community and the malaria research. Practically, I wouldn't say all the research, but the vast majority of financing and research that was done on malaria was made to do that. And indeed, each lab that that identified the gene considered it to be a candidate vaccine, and they all raced to try to vaccinate and to conduct trials with it. Now, the problem was that after 20 years of research and up till today, an effective vaccine that prevents the infection of malaria is yet to emerge. There is today only one vaccine that is able to reduce the likelihood of getting clinical malaria. Therefore, in a sense, the first lesson for the malaria community should be that cutting-edge technologies do not always fulfill their potentials. Now, the reason why they did not fulfill their potentials is because many of those people who came to do malaria research and to discover a vaccine ignored what we knew before. They ignored that acquired immunity is short-lived and it requires constant exposure, because by doing a vaccine, you're trying to imitate nature. They also ignored the fact that sterilizing immunity does not occur in nature. It was known that this was the case because parasite diversity had been uncovered. Different parasites elicit different immune responses. They also ignored certain things. They ignored that we did not know, and we still do not know today, exactly how immunity is acquired to malaria. Vaccination is a way to imitate immunity, and if you don't know how immunity is made, it's difficult to imitate it. We also don't know which antigens are involved in immunity, and we don't know whether the diversity of these antigens, genetic diversity, matters. There are, oh, sorry, pardon me. Okay, come on. There are other gaps in our logic for malaria, and these were difficult to study, even with the technologies that were available in the 1970s and 1980s, and I will not go through them now. The next phase and the next developments in biotechnology that have been helpful to malaria will be briefly summarized as follows. Genetically modified parasites were successfully made in 1993. Therefore, the application of genetic biotechnology to parasites only started in the mid-1990s. And up till today, only a few parasites, including Plasmodium falciparum, can now be manipulated genetically in order to either change some of the proteins by mutating them, mutating specific parts, tagging them with fluorescent proteins, and knocking some genes in and out, or making hybrid proteins where you take protein from one species and put it in the genome of another. This has opened the field of functional studies of malaria. You could actually now ask very specific questions to find out what each gene is doing and what its function is, which was very difficult and impossible to consider doing previously. The second major advance was the genomic era. Now, the genomic era, although it started with the discovery and the elaborations of sequencing, started in earnest when sequencing the whole genome became a feasible thing, a feasible endeavor. The human genome, for instance, took about 10 to 13 years in order to complete, starting from the 1990s, and cost billions of dollars. The Plasmodium genome is much smaller, 100 times smaller than the human genome, was sequenced for the first time in 2002 after about 3 or 4 years of effort. It was the first eukaryotic parasite to be sequenced, and within the same year, the first anopheline species, Anopheles gambia, which is a mosquito in Africa, an extremely good carrier of malaria, the genome was sequenced. We now have large numbers of genomes being sequenced today. The confluence of these three genomes, the three genomes of the three facets of the malaria parasites, has now made it possible to analyze in detail the interactions between these parasites. At least the possibility to do them, because if you know the gene sequences, you basically have a list of the proteins that exist and occur. It doesn't mean that you understand how they function together, but at least it's easy to get them and to manipulate them. For me, the development that is the most important and being the most useful for malaria was the development of in vivo imaging. The technologies that deal with in vivo imaging are evolving at an incredibly rapid pace. Seeing is believing, but seeing also how the parasite behaves, how the mosquitoes behaves in details and live is a fascinating and incredible thing, which was practically unimaginable about 30 or 40 years ago. The technology is so fast that actually it's difficult to follow how fast it evolves. I will how shall I say? I will show you a few examples that illustrate how fantastic this technology is. Now, these are few videos, and there are more for other parts of the life cycles that illustrate what we couldn't see before, and we can easily see now by using fluorescent parasites, for instance. This is the journey of the parasite from the mosquito. Am I still being heard?
Yes, yes, perfect.
Okay. This is the journey of the mosquito, of the parasites from the mosquito, the time the mosquito bites until it reaches the liver cell. So bear with me. Here is the picture of a proboscis of a mosquito biting the skin. This is the proboscis of the mosquito. And the green objects are the parasites that it injects from its salivary gland. It goes into the skin and deposits them into the dermis. As you can see, at each time it deposits a small number. It is not millions of parasites that are deposited in each bite, but maybe 15, 20, 30 parasites. The parasites are deposited in the derm, and here they are. In this case, they've been colored red, and one of them is highlighted because I'd like you to see what it is, what happens to it. As the parasite goes into the skin, they start moving around into the dermis, of course, until a time when they hit a blood vessel. And when they hit a blood vessel, you'd see what happens to it. Whoops, have you seen the parasite? It suddenly disappears and floats in. It goes and moves in the body until it reaches the liver. And here's what happens when it reaches the liver sinusoids. It crosses the barriers between the blood and the liver sinusoids. And here are the parasites in green again this time, moving around the sinusoids, finding from the red bloods, from the blood vessels into the sinusoids. And when they reach the liver, they are moving around, going through between. When they go red, it means that they're going through a liver cell. And they are going between and through liver cells, and they move around, penetrating and exiting liver cells until such a time that they find one in which they consider that it's a good liver cell for them, and they will start growing in it. I will speed up this video towards the end, where you can see the parasite has now settled in this cell, and this is where it's going to live. So the ability to visualize parasites has given the scientists an insight which would have been nearly impossible to get without that sort of technology. Oops, next slide. There have been many recent advances now today. Sequencing has become completely routine, and not only routine, but the new generation sequencing procedures, which are surreal in their efficiency and in their cost, have made it possible today to, for instance, sequence a human genome in a couple of days for a few $100s, considering that 20 years ago it took nearly 10 years and it cost billions of dollars. Transgenesis has been revolutionized by the CRISPR-Cas9, which allows you to very quickly manipulate the genome in ways that were unimaginable before. And as I said, in vivo imaging is generating a large amount of data and is becoming more and more complex. Unfortunately, also much, much more expensive to conduct than the usual microscopes that we have. We also had major advances in cytometry, flow cytometry, and mass spectroscopy, which when combined together are giving extremely detailed pictures of the immunological processes and the proteome of the parasites and of the human immunological cells. We can also now look at one sample and discover how many cells, CD4, CD8, all the variety of cells that are there, and measure cytokines, etc. It is generating a huge amount of data, and people are now studying parasites as a system biology, where they combine data from all these approaches in order to generate a picture of what is happening. This comes with its own problem. This is big data that is generated. Now we are talking about huge numbers of data generated from each sample, It is impossible to look at it without having a bioinformaticians and without understanding bioinformatics. This is, in a sense, the dangerous facet of the wonderful stuff that we can obtain from these things, is that now it has been, in a sense, the problem now is that we have so much data available that it's very difficult to extract meaningful, in some cases, meaningful information from it. The basic thing is that if you don't know the biological knowledge of your organism that you're studying, the biotechnology you use is just technology. So for those of you who go into the science, who are doing research in their organism, I would advise you to really study its biology before you use the technology that you have, because if you understand its biology well, your biotechnology will pay. And I leave you with this message, and I hope that I would hear from you and your advances and your successes at a later date. Thank you for your attention. I'm open to questions if you have any.
Right, thank you very much, Prof. Georges, for sharing with us the interesting research that has been done in Malaria and in a very early morning in France. Right, thank you, Prof. Right, so there's no Q&A sessions for keynote speaker, but if you have any questions you would like to ask Prof. Georges, you can type in the chat box. Yeah. Right. Next, we have another technical talk from the Impaco Private Limited Company. So Ms. Megan Bellows and Mr. Cheong are going to share with us their talks entitled Primer Dimers, Late CT, Pro Detections Limit, Say Goodbye to These Problems with PCR Biosystem. So Ms. Megan Bellows will be representing Impaco Private Limited Company, the sole distributors of PCR Biosystem. She is the Asia Pacific Channels Partner Managers at PCR Biosystem from the United Kingdom, with a vast experience in marketing and also the technical support. So without further ado, please welcome Ms. Megan and Mr. Cheong.
At PCR Biosystems, we're continually developing products to enhance your PCR, qPCR, and cDNA synthesis workflows. Our focus for this talk will be our qPCR Bio Green Mix, the dye-based qPCR product we've developed to help make this technique as efficient and cost-effective for you as possible. Dye-based qPCR uses a dye that intercalates with double-stranded DNA and upon doing so, emits a higher fluorescence intensity relative to when it is unbound. An excitation wavelength is emitted by the light source within the qPCR machine, and the dye's emission wavelength is measured by the fluorimeter through the FAM channel. At the start of the reaction, there will be small amounts of fluorescence because there are only small quantities of double-stranded DNA present. Once the PCR reaction begins, the DNA polymerases amplify the DNA template during each cycle, and the amount of double-stranded DNA present in the tube will increase. As the amount of double-stranded DNA increases, the more dye will intercalate with the DNA, thereby increasing the overall fluorescence. This change in fluorescence is measured at the end of each cycle, so the data is captured in real time, meaning quantification of your starting template amount is possible. When performing qPCR with the fluorescent dye, there are advantages and disadvantages to consider. One advantage is that you only need to design 2 sequence-specific primers for any one template, making it more cost-effective relative to the probe-based method. A disadvantage is that the dye will bind to any double-stranded DNA that is produced, including primer dimers and non-specific amplification products. A way around this issue will be discussed shortly. Let's take a look at our dye-based qPCR mix. Our green mix has been carefully formulated and includes a strategically engineered Taq DNA polymerase combined with antibody-mediated hot start technology. The antibody binds to the polymerase, rendering it inactive until the initial denaturation step. This prevents the formation of primer dimers and nonspecific priming and improves the sensitivity and specificity of the PCR. Antibody-mediated hot start is advantageous as it takes less time to denature the antibody compared to chemical hot start. Our mix contains a proprietary intercalating dye that does not inhibit the PCR reaction. Another feature of our mix is that it can be used to quantify any DNA template, including genomic, cDNA, and viral sequences. It is able to detect a low copy number of targets and overall exhibits market-leading performance, which we will see evidence of over the next 3 slides. We compared the performance of our mix to mixes from other manufacturers. 3 different target genes from mouse cDNA were amplified to ensure our mix was performing consistently regardless of template. A 10-fold serial dilution was carried out for every experiment, and reaction setup and cycling conditions were done according to the manufacturer's recommendations. The competitor mixes were tested in separate runs to ours to ensure each enzyme was performing under its optimal conditions. Our data will be shown in pink and the competitor's data in gray. The first row of data shows the qPCR amplification curves. The earlier the threshold cycle or CT value, the more sensitive the enzyme is. However, it's important to note that an early CT value doesn't necessarily mean the enzyme is doing its job correctly. It could also mean there are non-specific products present. This brings us back to the disadvantage of the dye-based detection method in qPCR. To ensure the correct product is being detected, a melt curve analysis is conducted at the end of the qPCR run. Our melt curves are shown in the second row of data. In a completely efficient PCR reaction, we expect to see 1 peak in a melt curve, and this means a single product was amplified. If there is more than 1 peak, then the reaction is not specific. PCR products should also be run on an agarose gel to verify if there is only 1 band at the expected size. The last row of data is the reaction efficiency, which was calculated by plotting the observed CTs against the log transformed template amount in each dilution. An acceptable value for reaction efficiency can fall anywhere between 90 to 110%. Efficiency values outside of this range indicate that the PCR product was not generated at the rate expected and that the reaction needs to be optimized. The qPCR amplification curves for the B2MG1 gene show our mix giving earlier CTs in all 3 cases. We are an entire CT earlier compared to competitor 1, and although competitor 2 has a CT close to ours, the MELD curve reveals 2 peaks, so there are non-specific products present, and this is likely causing the early CT. Our data is comparable with competitor 3, however, we have higher efficiency and still perform slightly better overall. This next set of data shows how we compare to other mixes when amplifying the ACTB gene. Compared to competitor 4, the MELD curve and reaction efficiency data look acceptable in each comparison of the mixes. However, the qPCR amplification curves show that our CT is roughly 3 cycles earlier. And while the CT values are almost the same as those of competitor 3, the MELD curves reveal that there is non-specific amplification taking place with the competitor mix, whereas we have a single peak. This demonstrates the higher specificity of our enzyme, which we attribute to our antibody-mediated hot start technology. Our mix has earlier CTs relative to those of competitor 5, and our reaction efficiency falls within the acceptable range compared to this mix. Our buffer chemistry plays an important role in the reaction efficiency. We've carried out stringent screening of the various concentrations of magnesium, enhancers, and additives to determine the optimal amount of each component that will best enhance our enzyme. This means little or no optimization is required for it to work efficiently with your samples the first time around. Something to keep in mind regarding the MELD curves is you may notice the position of the peak being different with our mix relative to the competitor's mix. This is normal because the differences in salt concentration and pH of each mix affect the melting temperature. The last set of data is for the ACTG gene. When comparing our results with the results from competitors 1 and 6, the MELD curves and reaction efficiencies are acceptable all round. However, what sets us ahead is the earlier CT values we can see on the qPCR curves. Our CT values are more comparable with those of competitor 2, and we both have one peak present on our MELD curves. However, it should be noted that their reaction efficiency is slightly lower than ours. Overall, some observations to highlight are as follows. All the data for our mix has consistently shown successful amplification of the template for all dilutions. This proves how sensitive our mix is. Our MELD curve data has consistently shown one peak, which demonstrates the specificity of our mix, and the reaction specificities in each instance have been within the expected range of 90 to 110%. Our mix can be used for various applications, including absolute quantification, relative gene expression analysis, and amplification of low copy number target genes. It's suitable for high-throughput qPCR from genomic, cDNA, and viral sequences, and can be challenged with complex templates that are GC-rich. Additionally, our mix has been tested under both standard and fast cycling conditions, and it is able to perform excellently in both instances. We've taken into consideration that researchers may be performing various quantities of qPCR within their projects. Our qPCR BioCygrine mixes come in a variety of pack sizes ranging from 100 reactions through to 5,000 reactions, each with low, high, or separate ROX options. In addition to our clear mixes, we have a blue mix which contains a non-reactive dye. The presence of this dye significantly reduces the likelihood of erroneously pipetting into the same well twice. The user will waste less of their time by being able to quickly ascertain which wells already have liquid in them. It's ideal when using small volumes or white plates. Our Cygrine blue mixes are also available in the same pack sizes as the clear mixes. Our qPCR mixes are compatible with all standard and fast cycling qPCR instruments. Our selection table will help users determine which ROX variant is suitable for their instruments. Our R&D team hasn't been the only team of scientists to observe the great results that our Cygrine mix brings. I've included some feedback from researchers at universities who have been more than satisfied with either the Cygrine mix or Cygrine blue mix. We also take great pride in our SciGreen mixes being used in research that has been published in journals such as Nature Communications, Molecular Cell, and Cell, to name a few. A more extensive list of citations can be found on our website within the resources section. Thank you very much for listening in. Impaco is our distribution partner within Malaysia and will be ready to discuss your PCR, qPCR, and cDNA synthesis requirements. They'll be available for any questions you may have.
Hi, good morning, everyone. Can you hear me?
Yes, we can.
Okay,
Yes.
sure. So thank you for the kind intention, and I hope you enjoyed the talk. All right. So here's a very important message from the two PCR experts and also the founder of PCR Biosystem. So here's what they say: we believe scientists deserve a better performance and value from their PCR products. So that's how they start developing cost-effective and high-quality PCR products. So you all can scan this QR code for more information or log into their website, right? So you can just screenshot it and then scan it later. All right. So in conjunction of this ICMBB, we would like to support students and also lecturers during this pandemic. So we are having this program called the Next Day Delivery Campaign. So I believe some of you are having headache to get reagents or consumables during the pandemic due to the shipment problem or delays. So if you want to know more about this, you can just contact us, or you can just scan this QR code as well. So this is another QR code which is linked to our website, right? So you can also screenshot this and then scan later, right? So this is my contact. If for more information, you can contact me anytime, right? Right. So thank you for your kind attention. Thank you.
Right. Thank you very much, Ms. Megan and also Mr. Cheong, for the interesting sharings, especially for the PCR's technical issue. So it's now come to the end of the first sessions, and there will be a concurrent sessions at Hall 1 and Hall 2 after this. So if you would like to listen to the talk on environmental biotechnologies and molecular biology, please stay at this hall. If you are interested on industrial and food biotechnologies and molecular biology, please go to Hall 2. To do so, you may click on the link in the program book to go to Hall 2. So we shall start our concurrent sessions at 10:25 a.m. Again, the current hall is Hall 1. So you may visit the respective hall now. Thank you. Hi, everyone. So welcome to Hall 1. This session is for environmental, biotechnologies, and molecular biology. So if you would like to listen to the talks on industrials and food biotechnologies and molecular biology, please click the link in the program book to go to Hall 2. So before we start, I would like to remind you again the house rules. So please mute your videos and audio. You will be given instructions if you need to turn on your video. If you want to leave the room and want to get back in, or you want to move to others' room, you have to click on the link in the program book. So if you have any questions to ask during Q&A sessions, please type your questions in the chat box and to whom it is addressed. Ladies and gentlemen, please let me introduce our chairpersons for these sessions, Associate Professor Dr. Chia Sui Lin. Dr. Chia, the floor is yours.
Right. Good morning, everyone. Thank you, Buchin, for the introductions. Without further wasting our time, let's start our first sections. Today we are very honored to have our first plenary speaker, Professor Hiroyuki Futamata from Shizuoka University, where he's affiliated with a Research Institute of Green Science and Technology, Faculty or College of Engineering, and also Graduate School of Science and Technology, and also Integrated Science and Technology. Professor Futamata obtained his PhD from Kyushu University in 1997. And he's an expert in environmental microbiology, specialized in bioremediations, wastewater treatment, biofuel, managing microbial community. He has published more than 50 papers and has been cited more than 1,000 times with the H index of 19. So I'm sure everyone is excited to listen to your talk, Prof. Prof. Futamata, your talk will be 30 minutes, and I'll ring the bell 5 minutes before the time is up. And again, like for the audience, please type your questions in the chat box and we'll collect them and ask Prof. Futamata during the Q&A sections. Well, without further ado, Prof, the floor is yours. Yeah, you may share your slide now.
Okay. Can you hear me?
Yes, we can hear you.
Excuse me, my presentation time is 10 minutes or 30 minutes?
Your talk will be 30 minutes, but 5 minutes before the time stamp, I will ring the bell.
Okay.
Thank you.
Okay. Excuse me. Okay, I start. Hello, everyone. My name is Hiroyuki Futamata, Shizuoka University, Japan. First of all, I appreciate to conference organizing member to invite me to a great conference held in Malaysia. Today, I would like to talk about mechanism of functional homeostasis in bacterial complex system. As you know, microorganisms play an important role for maintenance of environment and fermented food production and wastewater treatment. And recently, it's unveiling that the human gut microbiome are very related to our health. So in all situations, a microbial complex system is deeply related to the whole system functions. But we have not been paying the attention to managing the whole system function and microbial complex system, because a microbial complex system is naturally occurred. But I think it is very important to know how to manage the function of microbial complex system for developing the next biotechnology and constructing the sustainable society, because the a microbial complex system has a lot of potential. So firstly, I want to show the one of potential of microbial complex system, which is related to bioremediation. This research try to find a better way for efficient bioremediation about the TCE, trichloroethane contaminated environment. In this experiment, the natural soil and TCE was added in the bottle, and we measured the TCE concentration. Under the control condition, we prepared two styles of soil. One is sterilized soil and blue, not sterilized soil. And we incubated about 10 days. The TCE concentration, we find a little bit decrease, but in the, between the sterilized and not sterilized soil, no differences. It's meaning that this decrease not depending on microbial activity. So we try to enhance the soil bacteria activity. So we add a nutrient, but then the TCE was not degraded. So it is known that phenol degrader can degrade TCE by co-metabolic processes. So we try to enhance the phenol degrader, adding to be 20 ppm phenol, but the TCE degradation was not observed. So I thought 20 ppm phenol was not insufficient for enhancing the phenol degrader. So we increased the phenol concentration to 200 ppm. The TCE degradation, initially, I felt a little bit started, but then the degradation stopped. So we changed the approaches. We added the phenol to be 20 ppm phenol periodically 10 times. Under this condition, the phenol added, and then TCE was completely degraded. So according to this result, we can use the potential of microbial complex system when we manage them appropriately. So, but here is one question. Why could we succeed to make use of the potential? So to resolve this question, we try to analyze the phenol degraders. We collect the several kinds of phenol degraders, and then we check the TCE degrading activity, and unexpectedly, TCE degrading activity were very different. So someone is very low activity and someone is very high. And then we check the kinetic parameter for TCE and for phenol to understand the actual degrading abilities. And this kinetic parameter, positive correlation was observed between the Ks for TC and Ks for phenol. So these results, phenol degrader have diverse activity. In the natural environment, there are many kinds of phenol degraders. We have to stimulate only like TC high degraders among these situations. So how to stimulate TC high degrader selectively? So key point is high TC degraders exhibit high affinity for TC and phenol. So Ks value is low is meaning high affinity, and the Ks value is high is low affinity, is okay. So according to these results, my hypothesis is selective stimulation of TC high degraders is performed by addition of phenol at low concentration. It's a very important point. So to confirm this hypothesis, we try to make the chemostat culture. This is chemostat bioreactor constructed with soil as inoculum and phenol as soil carbon and energy source. As expectedly, the TC degrading activity is gradually increased and then reached to maximal level, which was highest TC degrading activity in the world. And until this, our hypothesis is correct. But when we measure the kinetic parameter of this reactor for phenol, we have another question. This number is sampling time, and then we check the kinetic parameter of this reactor for phenol. Initially, in the middle phase, the kinetic parameter is fluctuated, and then finally stable at high Ks values. Its meaning is low affinity. So chemostat culture can keep the substrate concentration at very low level in the culture. So we expect maybe the high affinity type bacteria can survive and then accumulate it. So the kinetic parameter position come here. But actually, this reactor's kinetic parameter stable at the low affinity positions. So this result suggested that there may be novel rule for managing microbial complex system with exception of kinetics. We're very interested in this phenomena, so we try to analyze microbial community structures. This figure shows a schematic diagram of bacterial community structure. As you can see, firstly, Pseudomonas dominated. After that, Acinetobacter and Ralstonia dominated. And finally, Varioblaxi dominated. So as you can see there, we found bacterial community succession occurred. So here we have two questions. Why cannot initially dominant bacteria, for example, Pseudomonas or Acinetobacter ralstonia, don't keep their dominance? And second is, how can initially minor population became dominant like this Varioblax? So these results and this question represent the feature of microbial complex system. This is a microbial complex system, and then this is microbial population. They have interspecies interactions. Some stress or environmental change, the system adapt to these changing with changing the community structure and interspecies interactions, like this. So this sequential changing is self-organization and community succession. And most important point is whole system function is maintained. So for making the best use of the potential, we need to understand the mechanism of function homeostasis. So to do that, if we use the practical actual environment sample, there is too complex to analyze. So now synthetic microbial community have much attention to understand the complex systems. This synthetic microbial community exciting tools to understand and apply microbial communities, because this SMC can reduce the complexity of natural ecosystems and also can increase controllability. So we try to make several kind of SMCs using isolated strains. Firstly, we try to make the synthetic microbial community using 2 strains under chemostat conditions. This synthetic microbial community kept the whole system function. So in this reactor, phenol accumulation was not observed, and also 2 strains coexisted. We try to add the 1 strain, so A+1. Unexpectedly, this synthetic microcommunity broken. Its meaning is sometimes graver meaning is accumulation of phenol. This synthetic microcommunity did not keep the whole system function. So we try to recover the whole system function. So we add the 1 more strain into this 3 strains synthetic microcommunity, so B+1, but we couldn't recover the function, the gray zone, meaning the phenol accumulation. So these results, all strains were phenol utilizer, but phenol was accumulated. So in this experiment, when we find the phenol accumulation, we stop the phenol supply. If we continue the phenol supply, these strains will wash out. So bacterial coexistence is indispensable for keeping the whole system function. So we need to understand the mechanism for bacterial coexistence for managing microbial complex systems. So we try to construct a new synthetic microbial community using the Pseudomonas LAB08 and Comamonas testosterone strain R2 and Capillabidus species strain P10. These 3 strains synthetic microbial community, this result is showing the monitoring of population density in the new SMC. And this blue bar, this is not phenol accumulation. This blue bar meaning the phenol supply stoppage. It's like a starvation to give the stress to the SMC. The running is very long, over the 800 days, but this SMC kept the whole system function. So never occurred, the phenol accumulation was not occurred, and then 3 strains coexisted. And most interestingly, for example, the phenol supply stoppage before in phase 1 and after phase 2. In phase 1 and phase 2, community structure, it was very similar. Its meaning is community structure shows the reproducibility before and after the phenol supply stoppage. So this MNC is useful for analyzing the mechanism of bacterial coexistence with functional stability of whole system. And we have 2 questions again. So why the strains LAB08 and strain R2 are simultaneously dominant? And why the strain P10 keep minor population? To resolve this question, we try to analyze the growth and kinetic parameter on phenol. This Ks value is affinity constant. Ki is inhibit constant value. The mu is a growth rate constant, as you know. The J value, this is survival power under the resource limiting conditions. this J value is calculated according to this equation. For example, a bacteria shows a very high growth rate. So mu is very, very high. And also this bacteria shows a high affinity for substrate. Its meaning is Ks is very low. Its meaning is very high affinity. Here is very large. Here is very small. Its meaning is this J value is very low. So low J value meaning is high survival power. So according to the J values, compare the strain R2 is maybe the overcome another strains because lowest J value. So we confirm this prediction using the mathematical model. This is Lotka-Volterra formulas. This is very simple mathematical model because this equation not including the interspecies interaction and heterogeneity of resource concentration. It's a very simple model. So calculation result is as expectedly, only strain R2 survive and then another two strains disappeared. But you already know in this SMC, all strains coexisted with functional stability of the whole system. So that this SMC was not formed according to kinetic and growth parameters measured on phenol. So there may be other factors for the coexistence. How did three strains coexist? So to resolve this question, we try to analyze the kinetic parameter of this SMC for phenol and catechol. Catechol is produced by phenol degradation. So until this, we only focus on phenol because this is first substrate. But from here, we focus on both phenol and metabolite catechol. This figure shows the dynamics of kinetic parameters for phenol and catechol of the synthetic micro ecosystem. This is strain R2 kinetic position. This is P10. This is LAB08. And the green spot show the kinetic positions of phase one. Phase one is before phenol supply stoppage. And after the phenol supply stoppage, this is phase two, is showing in blue boxes. Interestingly, this position is a little bit shifted. So in phase one and phase two, the bacterial community structure was very similar, but the functional equilibrium changed. These results suggested a hypothesis. So coexisting mechanism changed functionally. So we want to confirm this hypothesis. So we try to monitor the transcription level of these functional genes of three strains. So we try to analyze the genome of these three strains, and then strain R2 have one operon, including phenol hydroxylase gene and catechol 2,3 dioxygenase gene. The strain P10 has the same operon, and also strain P10 has catechol 1,2 deoxygenases separately regulated, and then LAB08 has a phenol hydroxylase gene and also catechol 1,2 dioxygenase gene, and these genes were regulated separately. So we check the transcription level of all these functional genes. This figure shows the transcription level of functional genes before and after phenol supply stoppage. As you can see, the strain LAB08, the pH transcription level was twofold than strains R2 and P10. And then catechol deoxygenesis gene transcription level was almost similar to pH levels. Okay, a little bit in strain R2 that this level is lower than pH, but I don't know exactly. And after the phenol supply stoppage, the in strain R2 was not so changed, but in LAB08 and then P10, the pH level is very decreased. And on the other hand, the catechol 1,2 deoxygenases gene level is very increased, especially in P10. This level was higher than 10 times than R2 and 5 times than LAB08. So bacterial coexisting mechanism changed before and after phenol supply stoppage. So summarize this data in the before. So the before phenol supply stoppage, all strain express the phenol hydroxylase gene. So according to this expression level, maybe this strain get to the phenol balance is normal, and then maybe 3 strains coexisted using phenol sharing. And after, in LAB08 and P10, the pH level is decreased, and catechol expression level is very increased. Maybe this is regulated by the catechol produced by strain R2. So, and then according to this expression level, the phenol sharing is in imbalance. So after situation, all 3 strains coexisted using metabolic networking system. So conclusion, so we try to understand the mechanism of functional homeostasis in bacterial complex system. So this mechanism is depending on the bacterial coexistence. This coexistence is depending on the flexible metabolic network, and this network depending on the diverse regulation of functional genes. So next challenge, we try to practical managing of microbial complex system. For example, try to redesign the microbial complex system. Of course, firstly, we try to using the synthetic microbial community structure. And to do that, we have to resolve the questions. Why, how did the coexisting mechanism change? So how is the metabolic network constructed? So to resolve this question, we focus on the interspecies interaction. This is one of the key important process. Now we're trying the analysis interspecies interaction. And question 2, the bacterial community structure was similar, but functional equilibrium changed. This is very interesting point. So how many equilibria does the SMC have? Maybe not only 2. Are these equilibria stable or unstable? This is stable dynamics equilibrium, but here is unstable point. How is changing? This is closely related to managing the microbial complex system. And question 3, metabolic network is key role in coexistence. So beyond our expectations, microbial metabolism may be incomplete. So for example, in my case, we try to isolate phenol degraders on the plate. Through this process, we got the isolate which have complete metabolic process, for example, from A to E. But in natural environment, I guess some bacteria only A to B, or some bacteria from C to E. So like this, metabolite leaking system forms microbial ecosystem. We want to show concretely, and then we try to redesign the microbial community. So I said acknowledge to my laboratory members, and then he is a Mauritian guy in PhD course student. We try to find a principle of functional homeostasis in microbial ecosystem and try to redesign for a sustainable society. And final, after coronavirus waves, I want to meet again in Malaysia. Thank you very much.
Thank you, Prof. Futamata. Really good timing, and that plate looks nice. Malaysian food. Well, I hope this pandemic finishes soon, and then we have more visiting from Japan, and we can go Japan as well. Really miss Japan. All right, so I'll keep the questions for the Q&A sessions later on, and I'll move on to our next speaker, which our invited speaker, Dr. Aklima, Associate Professor Dr. Siti Aklima Ahmad, and very old friends of mine. Okay, Dr. Aklima obtained her PhD from University Putra Malaysia in 2012 and continued to serve UPM after her graduation. And she's currently heading the Laboratory of Eco-Remediations Technology in the Department of Biochemistry, Faculty of Biotechnology and Biomolecular Sciences. Her research interests are in the fields of bioremediations, toxicologies, and environmental biotechnology and enzymology, focusing on tropical and polar environment, including Antarctica and Arctic. Right, I'm sure you have seen her pictures with all the penguins. And she has published more than 160 papers and with a H index of 19. So again, Dr. Aklima, your talk will be 20 minutes, and I'll ring the bell 5 minutes before the time is up. And for the audience, don't forget to type your questions in the chat box, so we'll collect them and ask Dr. Aklima during the Q&A sessions. Right, I'm not going to read her topic, and she will introduce the topic to you. So without further ado, Dr. Aklima, the floor is yours.
Okay, Hi, Dr. Eddie, you can hear me? Hello?
Yes, we can hear you.
Okay, I will share the slide. Okay. It's not here. Okay. Hi, a very good morning to all. My name is Siti Aklima Ahmad from Faculty of Biotechnology and Biomolecular Sciences, University Putra Malaysia. Today, I would like to present my research entitled Application of Antarctic Microorganism in Diesel Bioremediation Process. Bioremediation technique seems urgent and require more attention to solve the existing environmental problem. The pattern in the numbers of the publication published on the topic of the xenobiotic remediation of hydrocarbon degradation in the polar region, especially Antarctic, with the application of the indigenous microbial community has been increased from 2009 to 2019 before COVID. To address the state of the pollution, it is necessary to understand the mechanism and the process of the bioremediation of xenobiotic compound. the application of various microbial community originate from the polar regions can be provide a better interpretation on the mechanism of specific microbes in the biodegradation process. You can see the chart. This chart is summarize our Antarctic bioimmersion research. We are focusing on two microorganisms, which are bacteria and algae, and our main target pollution is hydrocarbon, such as diesel, engine oil, and waste cooking oil. Our second target is a phenolic compound. I have a collaboration with Prof. Tamata. For your information, most stations in Antarctic have a clinics, and they mostly use the antimicrobial solution, such as Dettol, to kill the germs in Antarctic. And then our third target is heavy metals and microplastic. Trace of heavy metal and microplastic have been reported to negatively affect to the Antarctic ecosystem. But today, I would like to present the data based on just based on the diesel degradation. This slide shows my experience in Antarctica for 3 years. I went to Antarctica in 2017, 18, and 19 to the two different stations, which is one is the Sydney Island, the British station, and the O'Hingie station, the Chilean base. Use of diesel. Diesel is extensively used to complement the human activity in Antarctica, especially for the scientific research station. Diesel oil is a petroleum product that supply fuel for the automobile, aircraft, and ship, as well as to generate the electricity in the station. Most research stations in Antarctica use a special Antarctic blend, SAB diesel, which is exclusive blended for the better performance in cold temperature. SAB has a lower heating value, is around 35,000 kilojoule per liter. Density is around 0.8 kilogram per liter at 15 degree. And SAB have content sulfur at maximum 10 milligram per kilo. Okay, and as encouraged by the Antarctic Treaty System, many Antarctic research stations are currently opting for the use of renewable energy source like solar, ocean, and wind energy. However, most stations in the region Antarctic still require embulsion of the diesel and other hydrocarbon to accommodate their power requirements. Okay, diesel pollution. The increasing rate of the accident involving vessel in the Southern Ocean, including the vessel sinking, has highlighted the potential for the substantial fuel spill into the Antarctic environment. The challenge in protecting the polar environment is the effective management of the hydrocarbon contamination emerging from the oil spill. Diesel pollution tend to be more persistent in the sub-zero temperature compared to those the warmer climes. It is due to the decreased velocity and evaporation at the low temperature, nutrient limitation that significantly inhibit the metabolic activity of the hydrocarbon degrading microbes. Okay, this is toxicity. More to toxicity. Policy, attitude, and awareness on the Antarctic on the environmental protection have improved in recent years. However, the risk of diesel pollution still remained. Okay, if we left untreated, the toxicity of the diesel will cause negative effect on the living organism in the polar regions. Okay, you can see this slide shows the several studies have been reported the toxicity effect of diesel fuel on the species richness, evenness, and the phylogenetic diversity on the Antarctic soil biota. It will inhibit the germination and early growth of the Antarctic plant, and also is affect the lethal or behavior on the Antarctic aquatic organism and animal, especially penguin and Antarctic seabirds. Move to the problem statement. The optimum condition of the diesel degradation by microorganisms such as bacteria and algae in Antarctica still undetermined. And then the diesel contamination is higher toxic to the marine organism, like I said, especially to the penguins. Okay, and then third, decontamination of the diesel is very expensive and time consuming because the Antarctic, there is no country in Antarctic. Okay, and then presently bioremediation has emerging as a superior and the most eco-friendly approach to removing contamination. Okay, you can see the photo. The photo shows the sampling activity in 2017 in Sydney Island, British Station. While on your right, in 2018 until 2020, the sampling was done in O'Higgins Station, Antarctic, O'Higgins Station, Chilean base. These are the methodologies have been used in the study. Okay, first is screening. So we're screening the best diesel degrading microbial community or the single bacteria. And then second, we optimize the best diesel degradation contamination using two methods, which is one factor at a time and the statistical using the response surveys methodology. And then after that, we identify the microbial community using the metagenomic analysis for the bacteria community, and also for the single, we use the 16S RNA and 18S RNA because we are just focused on the bacteria and also the algae. Fourth, we test the in situ experiment in Antarctica using the bioreactor system. Okay, this slide shows the first screening of soil sample, which is 1%. And it shows that 6 samples have an ability to degrade up to 80% of 1% diesel. And then go to the secondary screening of the soil. It shows that BS24 is the best bacteria from soil sample. While screening of the water samples, it shows that O2B have a high ability to degrade diesel at the concentration of 2% of salinity. Okay, and then in this study, after we select the best bacteria community, we did the optimizations. Okay, in this study, the bacteria community isolated from the soil, which is B24, successfully degrade up to 95% of 1.75% of diesel, while the marine bacteria community from O2B, okay, community isolate. O2B has the ability to degrade up to 80% of 1% diesel. Okay, and the bacteria community O2B has the ability to degrade diesel at the high salinity, which is 2.5% of salinity. Okay, this data obtained suggests the potential of Antarctic bacteria community in bioremediation application in the low temperature diesel polluted environment. You can see the temperature 12.5 and also 10 degrees Celsius is suitable to run the bioremediation experiment in Antarctic during the summer season. Okay. In early 2019, we did the preliminary in situ experiment has been run in the Antarctic based on the optimization data from the RSM from the RSM. Okay, and then in the mid 2019, our team has designed 2 machines for in situ bioremediation. First, you can see the jacketed incubator for diesel degradation, and you can see this machine have a motor, and the motor have a speed up to 120 RPM. Okay, and then the second machine is, we call it soil spinner. We use it to extract the diesel from the contaminated soil in Antarctica. Okay, same. We have a motor and the speed is around 100 and up to 120 RPM. In January 2020, okay, before lockdown, we bring the machine to Antarctic. We have a collaborator in Chile, and then still in January 2020, we also sent 2 PhD students in Antarctic. The in situ experiment have been run for 5 weeks in the Antarctic. Okay, first field activity. My student collect 10 points, have 10 point have been screened for diesel contamination starting from the tank. You can see the tank, the silver color, to the tank down to the sea. Okay, there is a 10 point. We call it 10 point. Okay, and then we try to extract using the gravimetric analysis. One sample only contain 200 grams soil. You can see from this table, it can show that B07 contain highest diesel, which is 0.657 gram diesel in 200 gram of soil. Okay, you can see BO1 until BO6 show the less diesel compared to BO7 to BO10. Okay, this slide, you can see the photos show the physical appearance of the soil. BO7 and BO10 is a little bit darker and oily compared to the BO1 until BO6. Okay, and then from this result, we use BO7 for using in the soil washer. This slide shows the effect of the soil washer period, soil washing period. So you can see from this graph, 2 hours is the optimum time for the soil washer, and it can extract up to 1.1 gram diesel. And then when we run the soil washer up to 7 hours, the total cumulative diesel weight is around 3.5 gram in 7 hours. Okay. And then we continue effect of the soil. The water ratio for washing in the soil washer, and it showed that 1:5 is the best ratio to extract the diesel using this soil washer. And then to speed up the diesel extraction, we use the STS, has been used as a biosurfactant in soil washer, and 1% of STS show the highest extraction of diesel. Okay, in the Antarctic, we just allowed to use the in bioremediation technique up to 1% of STS. Okay. In the future, we try to produce the biosurfactant from the bacteria, for the Antarctic bacteria. For in the future, we want to use the biosurfactant bacteria in the soil washer. And then lastly, we test the effect of the soil agitation speed. And then it shows that 40 RPM display the highest diesel extraction for our soil washer. Okay. Last experiment we did in Antarctic, we tried the effect of the jacketed incubator set up on the diesel degradation. As I mentioned before, the bacteria can degrade up to 1.75% 1.75% of diesel up to 95%. Okay. This experiment was run based on the optimized data from RSM. Okay, as I mentioned before, you can see the first 3 bar. Okay, the figure, the experiment will run based on the optimization based on the RSM. As mentioned before, we have this machine have a speed up to 120, and at the 120, it shows the degradation up to 75% of 1.75% diesel. However, when we add the bubble, we switch on the bubble and we still switch on the repeller, it shows that this bacteria can degrade up to 95%, the fourth bar, 95% of the 1.75 diesel. And we try to switch off the repeller, but the degradation is lower, it's less than 50%. So for conclusion, the degradation using the jacketed incubator is successful using the bacteria BS24. Okay, for details, maybe you can refer to my to our published journal. This slide shows the publication on the diesel degradation using the consortium microbial community from the soil and also the water. And then this slide shows that the single bacteria, we also isolate the single bacteria that have the ability to degrade the diesel in cold condition. Before I end my presentation. I would like to thank my collaborator from UPM, Dr. Noor Azmi, and also Dr. Suriana. For diesel degradation, we have a collaboration from international. We have a collaboration with the CIMA, with University of Mangallanes, also with British Antarctic Survey and Shibaro Institute of Technology, Japan. And we also have a collaboration in local from IMU, UITM, and also UST Science Malaysia. Okay. Last but not least, I would like to thank my student, my PhD and master student of Eco Remediation Technology Laboratory. Thank you. Terima kasih.
Thank you so much, Dr. Aklima. Thank you. All right, I'm sure you have a lot of questions to ask, especially, is the lab really cold? I'm always wondering, is the lab really cold? Because like she needs to grow the bacteria at like 10 and 12 degrees Celsius. Well, you keep the questions in the chat box and I'll ask during the Q&A sessions.
Okay.
All right, so ladies and gentlemen, we'll take a 5 minutes break. And when we come back, we'll start with our oral speaker. And before we move on, can I double check, is Dr. Roshan around?
Yes.
Thank you. Dr. Wan Irani?
Yes, sir.
Iriani? All right, thank you. Miss Rosina?
Yes.
Thank you.
Too.
Mr. Joel?
Yeah, I'm here.
Great. Mr. Shehu?
Yes.
Fantastic. All right, great. All of you will present live, right? All right, so we'll take a 5 minutes break. And when we come back, you can get your slide ready and I'll message you personally to tell you, okay, your time is up. Your time is ready. All right. So thank you very much. Toilet break for 5 minutes.
Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture.
Okay, we'll start our sessions now. All right, to all the speaker, your slot is 10 minutes and I will ring the bell. Sorry, my bell doesn't work, so I have to knock on my glass. So I will ring the bell at 8 minutes, indicating that you only have 2 minutes left. So be ready to wrap up your talk. Let's begin our first speaker for today. So today, our first speaker will be Associate Professor Dr. Roshan Mascarenhas from Newcastle University Medicine. His talk will be on the bacterial diversity analysis in mangrove ecosystem of Johor using Minions sequencing. Right, Dr. Roshan, the floor is yours.
All right, I'll share my screen. Can you see my screen?
Yes, we can see your screen. You may start now. Thank you.
So good morning, everybody. My name is Roshan. I'm from Newcastle University Medicine, Malaysia. So I'm presenting today our work on bacterial diversity analysis in mangrove ecosystem of Johor using MinION sequencing. So you're all familiar, those who are from Malaysia, about mangrove ecosystem. So they are the coastal ecosystem found mostly in tropical, subtropical regions, and their bed with the sediments provide a very rich habitat for diverse microorganisms such as bacteria, fungi, etc. And due to varying the habitat conditions such as salinity and pH, the organisms that survive in this environment needs great adaptation. And therefore, the organisms with distinct features can be found in these sediments. However, these days, we all notice that mangrove vegetation and ecosystem has been depleted due to increased human activity. And it is sad to know that there are many microorganisms in this habitat still to be understood. And one of the challenges is to culture these microorganisms because we cannot imitate the same habitat situation in the lab. And that's why many are still being under investigation. So one of the ways to identify the different bacterial diversity is by using culture-independent molecular methods that will give the idea about different bacteria in that community. Therefore, we sought to explore the overall bacterial composition, abundance, and diversity in the mangrove sediments. For example, actinobacteria, which are actually very important in terms of their biomolecules. They are adapted to various ecosystems, including mangrove, and they are very important because they are the rich source of biomolecules such as antibiotics. And in mangrove vegetation, you can see these are challenged continuously by different salinity, and therefore there is a chance that the bacteria with unique abilities could be present in this environment. And with that in mind, so we had two objectives in our mind: to explore overall bacterial composition, abundance, and diversity in the mangrove sediments of Johor, and to identify bacterial diversity in differentially exposed mangrove vegetation, such as with the less human activity and more human activity. So our workflow included sample collection. We collected these soil sediments from locations of mangrove in Sungai Malayu area and Pontian region, and which are differentially exposed with the human activities. And then a portion was used for bacterial culture and backup. Our main aim was to sequence DNA directly from these soil sediments. What we used was isolated DNA using fast DNA spin kit columns. And once the DNA isolated, we prepared library for MinION sequencing. We sequenced 16S rRNA gene for all this DNA, and then taxonomically identify which group they belong. And after barcoding, so we pulled these samples and then used sequencing, where we used base calling, and further processing of data was done on software called Epitome. And this is a MinION device. For those who don't know, this is a handheld instrument sequencing device which comes with different kits. So we use for 16S rRNA kit. You can also use for transcriptome and genome sequencing. And what comes unique is this flow cell, which is specific and which is actually use and throw after 1 or 2 runs. And the sample is loaded. It has been prepared in previous slide in this sample port, and then just connect it to the laptop and run the sequencing run overnight using this system. So what we get at the end is the reads, and the reads are then using the softwares that classify the reads. 88% of the reads are classified. That means they can identify a particular type of bacteria, and 12% are unclassified. Probably this 12% unclassified comes from spurious amplifications, as well as those bacteria that are still not classified or discovered. So majority of the bacteria phyla, as in any of the mangrove vegetation, so Proteobacteria, Bacteroides, followed by Actinobacteria. So we found this pattern of like Proteobacteria, Actinobacteria, Bacteroides dominating in these ecosystems in our sequencing. This is a sample of like different types of phylums that are identified. Nearly 16 phyla are identified, and this is a Proteobacteria, shows diverse species in this particular phylum. So to understand better, I have classified like this. In location 1, it is pristine and mariculture exposed. The pristine is in Kukup, where it is well protected, and some regions of the periphery are exposed to mariculture. In location 2 is upstream of Sungai Malayu and downstream. Downstream is heavily polluted, and upstream is slightly disturbed due to development in Iskandarputri region. So the results shown here compare the diversity of phylum, class, and genus between different mangrove sites. So shown in blue is the phylum. Across, you can see there is not much difference, though there is 1 or 2 phyla reduced in polluted areas. Whereas class also, there is not much difference. When it comes for genus, so this is in the pristine, you have highly diverse bacterial population, and which is exposed to mariculture is slightly reduced. And then in upstream of river, which is already slightly exposed to development, there is a reduction compared to pristine. However, compared to the same location downstream of Sungai Malayu, which is heavily polluted, so you can see reduced number of significant reduction in genus. So further, we also found the specific type of bacteria enriched in different systems. In pristine and upstream, there is a wide variety of Erythrobacter that is dominating genus, and number of species, whereas in downstream and mariculture exposed areas, it is reduced, depleted Erythrobacter, where Illuminobacter, where only 3 species are coming predominantly high. So one is Proteobacteria, other is Actinobacteria. What this shows is there is a tip of balance between the diversity of bacteria which is exposed to human pollution and activity, and reducing the diversity of bacteria in that environment, and it favors some organisms which can adapt to that environment to grow and survive. To conclude, so biodiversity of soil microbes is very useful as it provides the indication of ecosystem change, and this study showed that there is reduced bacterial diversity due to human activities in mangrove. And these studies are important as they create a global impact and help to conserve ecosystems, mangrove ecosystems. And MinION sequencing is a powerful tool to detect those microbes that are otherwise very hard to grow in labs. So our future direction is to sample more areas and validate our findings, and also look for bioprospecting to identify actinobacteria with the unique properties, biological properties. So I would like to thank Dr. Madiha, our Dean, and our CEO, Prof. Baldwin, and research fellows, and Dr. Jayanti, our Assistant Dean of Research, for their support, and NewMed for funding this project. Thank you.
Right, thank you, Dr. Roshan. Very good timing. Thank you so much for keeping it that way. All right, so we'll have our next speaker, Associate Professor Dr. Wan Iriani. Her title will be Eco-Friendly Silver Nanoparticle Fabricated by Green Synthesis, Mafisha Mobidi. Dr. Wan Iriani.
All right, thank you, Chairperson. So I would like to share my, my slide now. Can you see my slide?
Yes, now. Thank you.
All right, thank you to the Chairperson. So my name is Wan Iriani Wan Ismail from University Malaysia Terengganu. I would like to share our research about eco-friendly silver nanoparticles fabricated by green synthesis using marine polychaete, Mafisha Mobidi. So according to WHO, in World Economic Forum in 2020, actually, in addition to COVID-19 pandemic, actually we are having 7,000 signals of other potential outbreaks every month, actually. So it is indicating that more infectious diseases will be occur in the future. Because of the problem, definitely we need to look at a new antimicrobial agent in order to combat that all types of infectious diseases, including antibiotic, antiviral, and so on. Unfortunately, we're still having antibiotic resistance in order to combat related diseases. So that's why some of scientists now, they are looking to the new alternative to produce antimicrobial agent from nanoparticle. So in our study, we would like to share how to synthesize one type of nanoparticles known as silver nanoparticles. And basically they have 3 methods to synthesize silver nanoparticles, including physical, chemical, and also biological method. But in our study, we have preferred to focus more on biological approach because they are more eco-friendly and also considered as green technology. So once we talk about silver synthesis of silver nanoparticles, we should know that silver nanoparticles, they have a very specific size within 1 to 100 nanometer. Because of their very small size, indicating that the nanoparticle have large surface to behave as antimicrobial agent. At this moment, the value of the nanoparticle is very high, as getting triple because of the COVID-19 pandemic, because some scientists believe that the silver nanoparticle can become as one of solution in combating COVID-19 pandemic. So in this study, we are focusing more on green synthesis using biological methods. So they have a few types of biological specimens that can be used in synthesis silver nanoparticle. But in our study, we are focusing more using marine organisms. So we use specifically polychaete, or also known as marine worm. This marine worm, locally known as a rod bakau. You can see the figure of the marine worm. And at this moment, we use this marine worm as a fish bait, and we can get this kind of marine worm from mangrove area. Actually, we use this polychaete for other studies, and from the study we produce a lot of waste from the polychaete. So from the waste we found actually we can reuse the waste of the polychaete to produce silver nanoparticle. So because we already know that silver nanoparticle is a very excellent antibacterial have an antibacterial activity. However, we don't know yet how this polychaete can synthesize silver nanoparticle and how the antibacterial activity from the synthesized silver nanoparticle behave. So that's why we want to conduct a study to synthesize and to optimize method to synthesize silver nanoparticle from the polychaete. Then we can characterize the structure, and before we can do the antimicrobial activity. So this is briefly our method that we use for this study. We collect the polychaete, we do the acclimatization procedure, and then we optimize the synthesis based on the 4 categories here, including class of the polychaete, storage of temperature condition, silver nitrate concentration, and also pH of the crude extract. After that, we can characterize the optimized silver nanoparticle using the 5 methods before we can do the antimicrobial activity. So briefly, I'll show you how we extract the polychaete. Then basically, we need to add silver nitrate as a precursor to reduce to produce silver nanoparticle from the polychaete before we can observe the color changes, and also we need to use UV-based absorbance spectrophotometer to observe the silver nanoparticle formation. So from the results, we found based on this 4 category of the optimization procedure. So based on the color changes, we found that class 2 of the polychaete showed a better result compared to other classes. We also compare our study with commercial silver nanoparticle from Sigma. We also compare the study with the crude extract without silver nanoparticle as a precursor, and also we also compare the study with silver nitrate without any reducing agent. So we confirm the color changes based on the UV-based spectrophotometer from 5 hours up to week 4, and we can see the consistent peak formation here. This is the black color of the peak, indicating to the positive control. Our sample is in the red, green, and also blue color. This is based on the different classes of the polychaete. From this study, we found that class 2 of the polychaete indicated the best silver nanoparticle formation compared to other classes. And then we also measured, we also optimized the temperature, how to store the silver nanoparticle, and we found that temperature at 4 degrees was the best temperature to store the silver nanoparticle, which we can stabilize the silver nanoparticle up to 6 months. And then we optimized the concentration of silver nitrate to be used in this study. Based on the study, we found that 0.5 up to 1 millimolar was the best concentration of silver nitrate to be used to synthesize the silver nanoparticle. It was confirmed by this spectrophotometer peak. And the last part of to optimize the silver nanoparticle synthesis, we also observed that the best pH can be used in the study. From the study, we found that pH 9 showed the best result to synthesize the silver nanoparticle. So what we can summarize from the optimization procedure, we found that polychaete from class 2, that's indicating class from the adult stage of the polychaete, was the best class to synthesize the silver nanoparticle, that we need to store the silver nanoparticle at the 4 degrees to maintain the silver nanoparticle. And we also need to use silver nitrate at 1 millimolar at pH 9 to synthesize the silver nanoparticle at the best condition. After we manage to optimize the condition, we need to characterize the silver nanoparticle using 5 different methods here. By using the dynamic light scattering DLS, from this screening method, we found that silver nanoparticle in 91.21 nanometer in size. But definitely we need to use SEM and also TEM to confirm the size of the nanoparticle. Based on the SEM, we found the silver nanoparticle in the spherical light structure. And we confirmed the size of the nanoparticle actually was 40.19 nanometer. And also we confirmed the shape of the nanoparticle in the spherical shape form. And we also did X-ray diffraction or SRD to confirm that silver nanoparticle that we synthesized from the polycate, originally from the polycate itself and not from the environment. And so we confirmed that our result is solely from the polycate, not from the contaminant. And then we also measure what the specific organic compound involved to synthesize the silver nanoparticle. And basically we found that a few organic compounds involved in the synthesis, including carboxylic acid, phenols, alkalines, aromatics, and also amines groups. And a summary of the characterization. And lastly, once we did the antimicrobial, especially as an antibacterial study from the 8 types of 8 species of bacteria, and we compare with the positive control and also silver commercial silver nanoparticle from Sigma, we can see that our silver nanoparticle can behave as antibactericidal to the 7 types of microbe in this study. So as a conclusion, we can confirm that we can optimize and also characterize the silver nanoparticle synthesized from the polycate. And then this synthesized silver nanoparticle show the best result on the 7 species of microbes. And definitely at this moment, we are looking forward to study more in detail about the molecular mechanism to understand the mechanism of action of silver nanoparticle. That's all from me. You can get more information from our publication. And that's all. Thank you.
Thank you, Dr. Wan Iriani. Sorry, because I know it's very, very difficult to present all you have very fantastic result there, very interesting. But we have to move on to our next speaker to keep the time. All right, our next speaker is Miss Rosina Kaka. She'll be presenting her topic, preliminary synthesis of P something using glycerin pitch as a main carbon source from the bacteria, USMAA 1020. Rosina, the floor is yours.
Hello, am I audible?
Yes.
And
yes,
can you see my screen too?
Yes.
All right, so Assalamu alaikum and good morning to everyone. This is me, Rosina Kakar from Pakistan, and I'm doing my master's research in microbiology from University Sains, Malaysia. And it is an honor for me today presenting my research work among the great researchers all around the world. So I'm really grateful for this opportunity, basically. Today I'm presenting about the preliminary synthesis of the poly-3-hydroxybutyrate, copolymer 3-hydroxyvalerate using glycerin pitch as main carbon source from Cupraoideus melanchiensis USMAA 1020. Basically, this bacteria is locally isolated from the Pulau Pinang Lake in 2008 by Prof. Amiral. Talking about background of my study, like from many decades, global efforts have been made to replace the petrochemical plastics over the biodegradable plastics and environmental friendly plastics because of the fact that the petrochemical plastics can take many years to decompose in the environment, and also it is a threat to our environment because of its serious consequences. So scientists have found basically a natural biodegradable polymer which shows the same properties like the plastics, and it can synthesize and can formed from the microbes, so mainly from bacteria, which is called as polyhydroxyalkanoids. So now the major challenge in the PHA research that the scientists have found that its production can take, it's very costly, the production, if you see. So now they are shifting their research area to use the raw materials, the cheap raw materials like glycerin and glycerin pitch. So glycerin pitch is basically a byproduct of the biodiesel industry, and biodiesel production can take place through the transesterification process. So there is since glycerin pitch, it's largely produced in Malaysia, and there is only one body, which is the Malaysian Quality Management Waste Management body. It is only doing the handling and treatment of this waste, but it is also taking charges. Like for landfill, it can take 510 ringgit, and also for the incineration process, it can take from 810 ringgit till 3600 ringgit, which is costly. And it is also like, why not before, since most of the glycerin pitch is dumped into the soil, why not taking advantage of taking out the valuable components from the glycerin pitch, because it has usage in many industries as well. So this is a rough illustration talking about the economic sustainability in the PHA industry, in which it is showing that from the waste to wealth, from glycerol and glycerin pitch, production of the PHA can reduce the petrochemical plastic pollution, and also can reduce the raw materials, can reduce the waste generation, and also can decrease the cost production too. So objective of this study to synthesis of biodegradable poly-3-hydroxybutyrate copolymer 3HV from the Cupravidius melanchiensis USM AA1020 using glycerin pitch, and to optimize the production of copolymer 3HV through shake flask fermentation, and to test the different parameters in order to achieve the high pHA content and its monomeric composition. In methodology, I have done the biosynthesis of this polymer using glycerin pitch as main carbon source through the shake flask fermentation, and at around 200 RPM, and for 48 hours, in which I have done the first experiment to check the effect of different concentration of the glycerin pitch, and then I moved towards the effect to check the effect of different precursors, and in which I have concluded that 1-pentanol is the very suitable precursor for my polymer in order to produce the PHA. Also, I have checked the effect of different concentration of ammonium sulfate, and in the last, I have run the fermentation over the different concentration of oleic acid. Introduction of the oleic acid was to enhance the PHA production. So all of these experiments were carried out in triplicates, and the cells were transfugate after the harvesting at around after 48 hours, and then the cell pellets were freeze dried, and then I have analyzed it in gas chromatography, leading with the methanolysis to check the monomeric composition. In results, for the glycerin pitch, different concentration of the glycerin pitch, I have checked that with the increase of the glycerin pitch concentration, I have observed the increased rate of the PHA production too. And in the next experiment, talking about the different precursors from 1-pentanol, noneic acid, propanoic acid, and valeric acid, again, I have noticed the high optimized condition of the PHA content, and also for my copolymer 3HV with the 1-pentanol concentration. And again, once finalized the 1-pentanol precursor, so I have tested the different concentration of 1-pentanol with the weight % carbon. And And moving forward for the salt that I've used in the medium, I have also played around with the salt concentration to check that is it the salt really affecting the PHA production, because for bacteria to utilize, like it can grow in the minimum amount of the salt it can produce the PHA or PHA. So basically I have found that it was 1.1 was more likely giving me the accurate PHA content. And while I didn't see the significant effects of the rest of but still high salt intake cannot be so good for the bacteria PHA content. So yeah, the last experiment, which was for the oleic acid, again, I have done with the different concentration of the oleic acid for 48 hours. This experiment was basically to boost up the PHA content, and where I have found the oleic acid till 1%, I have seen the increase of the PHA content. But still, like if I go beyond the 1% weight percent carbon of the oleic acid, it can still slightly dangerous to the bacteria. So yeah, with this all experiments, I've concluded that the effect of different concentration of glycerin pitch 1-pentanol ammonium sulfate on the production of PHA by Cupriavirus melanchiensis USM AA1020 was investigated. In results, Cupriavirus melanchiensis USM AA1020 was selected with the P3HB and copolymer 3HV from the glycerin pitch and 1-pentanol. So with the conditions of 20 gram per liter glycerin pitch, 0.06 weight percent carbon for 1-pentanol, and 1.1 gram per liter ammonium sulfate for incubation period of 48 hours, and for 200 RPM in shaker. So I'm still this project is still ongoing and I'm still looking forward for scaling up the production of the PHA in bioreactor, and also I will be working on the characterization of this polymer. And for bioreactor, I'm using a software, Expert Design, in which I'm studying the response surface methodology for optimization process. So thank you so much for paying attention towards my presentation. Yeah, that was all from my side.
Thank you, Rosina. Very interesting finding. Wish you all the best for your study.
Thank you so much.
So,
thank you.
yeah, well, up next is Mr. Joel Zakaria from UCSI. All right, his title will be Immobilizations of Oil-Degrading Bacteria and Cellulose Triacetate beads formed from carport fibers and their role in oil spill remediations. Joel, the floor is yours.
Thank you for the intro. Hello everyone, my name is Joel Zakaria, and my topic is the immobilization of oil-degrading bacteria on cellulose triacetate beads formed from carport fibers and their role in oil spill remediation. Okay. So when we talk about oil spills, it's actually a pretty good tongue twister. You can try it. You can say oil spill, your tongue will get twist for sure. Anyways, most of you will recall the oil spill that occurred in Mauritius a few months ago, or if you're a bit older, you remember the Deepwater Horizon spill about 11 years ago. Despite the awareness, oil spills are still recurring, and in Malaysia, oil spills go unreported, especially those on the Straits of Malacca, one of the busiest straits in the world. Oil spills consist of a large number of hydrocarbons, including polycyclic aromatic hydrocarbons, or PAH, which are harmful compounds to humans. The effects of oil spills don't just vanish overnight. In fact, the Deepwater Horizon spill that occurred 11 years ago still affects sea animals and the ecosystem still today. Remediation of oil spills is a legit concern due to how often it happens. And in Malaysia, the recent oil spill that occurred was cleaned using diapers. That's right, diapers. The problem with this is that diapers don't just absorb the oil, they also absorb the water as well. Not only that, but what happens to diapers once they've been used? Most diapers aren't biodegradable, and it's not very sustainable in it. And furthermore, dispersants used in other countries are also quite expensive. So let's take a closer look at the biological method to clean up oil spills, or what we call bioremediation, like the previous talk was talking about. And so there are actually various types of microorganisms that use hydrocarbons as their sole source of energy. However, due to waves and currents, these microbes tend to get their food source separated from them. Pretty much like, can you imagine eating your favorite burger and getting slapped in the face? That's pretty much how the bacteria feels. Because of this, could we give those specific microbes something to latch on to? while consuming the hydrocarbons. So the question came, what if you could combine this mechanical method where a solvent is used to absorb the oil and the biological method where bacteria degrades the oil. Kapok is an indigenous crop highly valued for kapok oil. It has been proven to have high absorbency of crude oil because of its hydrophobicity. Immobilization of a bacteria on an inert material like kapok opens up the possibility of recovering and reusing the solvent multiple times, making this a sustainable process. It also reduces the amount of secondary waste and basically the use of one-time solvents. Thus, this is my current plan for the whole project. Most of it is almost done. I mean, most of it is done. I only have a few things left to do in this project. So basically, the first part of the project was to modify the couple fibers, and to modify it meaning to make it more hydrophobic. If you don't understand these terms, later on I'll explain. So yeah, the first part was to modify it, and the modification process changed it into powder, and then tested the degree of acidification. And from the powder, we wanted to make something out of powder, so we made beads out of that fiber. And from there, we tested the absorption capacity of the beads, and we mobilized the bacteria on the beads. And the next part would be the reuse cycle part 2. And the last part, which I haven't done, is the oil degradation of the oil. All right, so yeah, like I said, the first part of the project was to modify these naturally water-repelling couple fibers and make them more water-repelling. I achieved that through a process called acylation. So just remember that higher acylation equals higher water repellence. To date, the acylation of fibers shows that unprocessed couple fibers had an acylation value of 0.0085, and the processed couple fibers, which are the powder on your right, have a value of 2.88. But as you can see, the fibers are no longer fibers, they are now a powder. So not only did we measure this through titration, this value through titration, but we also ran the samples through FTIR. And you'll see the FTIR here. So yeah, on the top is the untreated fibers, and the bottom is the powder or treated fibers. So as you can see, the hydroxyl group is the main chain on the fibers. And basically, once we treated them through acylation, it completely displaced the hydroxyl group with the acetyl group. And those are the values for the acetyl group right there at the bottom. Okay, so basically, like I mentioned, they weren't fibers anymore, and they were just a powder. So we were thinking of what could we do with this powder, and the idea is to make them into porous beads. So we made 4 types of beads by manipulating the cellulose to solvent ratio and manipulating the precipitation solution. So all these calculations are pretty complicated. I mean, not complicated, but they're tough to explain right now. So if you have questions how to calculate 12.5 and all that later. Okay, so we did the absorption capacity of the beads. We compared all 4 beads first. So as you can see, the 16.67 methanol and 12.5 methanol didn't do as well as the 16.67 DH2O and 12.5 DH2O. So basically, the following parts of the experiment, we only used the 16.67 and the 12.5. Although the 16.7 is slightly lower than 12.5, but yeah, there's a significant difference between all 4. All right. Next part is the reusability of beads. Like I mentioned, we continue to test using 12.5 and the 16.67. So as you can see, the reabsorption ratio maintained as an almost straight line for the 12.5 and 16.67 as well, but the absorption capacity was higher for the 12.5 compared to the 16.67, which is why we did the immobilization of bacteria only for the 12.5, not the 16.67. So next up, you'll see the fibers. We did a reusability test for the fibers as well. As you can see, the reabsorption ratio drops really far compared to the beads. All right, so we viewed the beads under SEM. So you see the 12.5 and the 16.67 beads. So yeah, on your left, you see the 12.5. On your right, you see the 16.67. And that's the view from the outside. And this is what they look on the inside. So as you can see, the 12.5 is much more porous compared to the 16.67, which could explain why they have a higher absorption ratio. Okay, so the next part, like I mentioned, was the bacterial immobilization. So we chose a specific bacteria, Acinetobacter veniceus, to be immobilized on the fibers and cuphook. So yeah, we managed to immobilize the bacteria on the fibers and the cuphook, and we managed to get a CFU of 3.05 on the fibers and 1.32 on the beads. And like I mentioned earlier, again, we used 12.5 beads for the immobilization. So we also view that under SEM. So as you can see, this is the surface of the beads, not the fibers, because we're supposed to do the fibers as well, but because of MCO, we couldn't. So basically, as you can see there, the bacteria is right there, all these small little cute little dots. Those are my bacteria, Acinetobacter veniceus. And yeah, so the next part of my project would be to compare the immobilized bacteria and the free-floating bacteria. So to summarize everything, in theory, the process, couple fibers absorb oil, and then the bacteria attached to the fibers consume the oil, which ultimately makes the beads and the fibers reusable again. So it is an ongoing project, and we are super excited to see what outcome yields. Thank you for your attention.
All right, thank you, Joel. Thank you so much for your interesting presentations. All right, so we'll move on to our last presenter for today, Mr. Shehu Idris from USM. His title of presentation will be Bioprospecting and Molecular Identification of Waste Transformers of Oil Degrading Bacteria for Bioplastic Productions. Shehu, you can share your slide now.
Okay. Yes, can you see my slide?
Yeah, we can see your slide. Fantastic.
All right, may I proceed?
Yes, please.
All right, good afternoon, everyone. My name is Shehu Idris from University Science Malaysia. I will be talking on the topic bioprospecting and molecular identification of waste transformer oil degrading bacteria for bioplastic production under the following outlines. As an introduction, huge amount of plastic waste are generated daily from the usage of non-biodegradable plastic. And this has become a major environmental issue because most of the plastic being used are from petrochemical base and they take very long time to degrade in the environment and creating a serious environmental issue and problem. Although it is hard to completely stop the use of non-biodegradable plastic, their versatile utility, it is, however, possible to substitute or reduce their usage with better alternatives. Fortunately, microorganisms are capable of converting several waste value-added products, for example, polyhydroxyalkanoate. Utilization of waste transformer oil to produce value-added products will help in the reductions of the problem associated with disposal. The scope of the research, the idea is to look for a cheaper carbon source for the production of these eco-friendly materials, partly because productions of biodegradable plastics associated with huge production costs. So getting a cheaper carbon source for the production of this material will really help in the commercialization of this important material. The aim of the research is to isolate and identify bacteria capable of utilizing waste transformer oil degrading oil as a carbon substrate for the bioplastic production, while the objectives are: one, to isolate the waste transformer oil degrading bacteria; two, to assess the waste transformer oil degradation potential of the isolated bacteria; three, to screen for the polyhydroxyalkanoate producing bacteria using waste transformer oil as carbon substrate; and finally, to identify the PHA producing bacteria using molecular approach. Here is the experimental flow chart itemizing the steps involved in the isolations and subsequent analysis, starting from the isolation and the screening via enrichment, serial dilution, and isolation of the pure colonies using separate plate techniques, and screening of the PHA using near-red staining and fluorescent microscopy, and confirmation using transmission electron microscopy and gas chromatography, and the oil degradations using solvent extraction and gravimetric method, then the molecular identification through genomic DNA extraction, amplification of the target DNA sequencing, and the analysis of the sequence. To achieve the isolations, 3 different samples were collected comprising soil, wastewater, and even the transformer of oil. The sampling site indicated, including Penang Barung down to Batu Feringhi, all here in Malaysia. So to isolate the bacteria, we started with the enrichment of our culture. Then the mineral salt medium was supplemented with 1% volume over volume of the waste transformer oil. We are very careful in adopting a higher concentration of the waste transformer oil because of the toxic nature of the waste transformer oil. And eventually, the bacteria were successfully isolated for the subsequent analysis. Then the next, we move on to the oil degradation analysis, in which we adopted the solvent extraction method with N-hexane as the extraction solvent, a culture of the organism in the mineral salt medium, but this time around with the 2% oil, because we are targeting as getting the residual oil that can be used to compute the percentage oil degradations as indicated in the formula. Next, we move to the PHA biosynthesis and quantification. Mineral salt medium was used, and after incubation for 72 hours, we recover the biomass, freeze dry, and carry out metanolysis. Then finally, we compute the PHA content. The culture condition and the GC parameters are indicated. The molecular identification, first through the normal conventional methods, through the isolation or extraction of the genomic DNA, the PCR amplification of the target gene, which is 16S-RNA gene, then clean up on the sequencing of the result, the PCR parameters indicated there. The result here is showing the abundance of the used transformer oil creating bacteria isolated from the different sample. There we have a total of up to 2 different morphotypes of these organisms recovered from the different sampling sites. Here is showing the plate of some isolated organisms, showing different morphology and pigmentation in some cases. Next, we have the result for the screening of PHA productions. In this result, we screen those 62 isolates that are able to degrade waste transformer oil, out of which we got up to 16 isolates that are showing a sign of PHA accumulation. From the other side, figure 3 shows the plates showing the appearance of the fluorescent microscopy of the 9 red stem cells, then the surface morphology of one of the selected organisms and the phase contrast, as well as the transmission electron microscope for the confirmation of PHA granules. Then we move to the quantification of the oil degradation and the PHA. From this result, we can see, as highlighted, the different isolates with their code here were able to degrade oil in the range of 19.58 down to 57, up to 57.51%, while the PHA content as determined using gas chromatography range from 0.6.59 up to 33.06 weight percent, with a different cell dry weight in gram per liter. Here is showing some plate indicating the polymer we make attempt to extract and analyze using gas chromatography, while in the other C and D plate is showing the ionization impact obtained from the gas chromatographic mass spectrophotometry, one of the selected isolates. For the molecular identification of the result, after successful amplification of the 16S-RNA gene and the sequencing, the sequence obtained were assembled and refined. Then we compare our sequences with the sequence in the NCBI database, and here are the possible organisms identified, comprising of Acinetobacter, Serratia, Proteus, and Bacillus. Here is showing the agarose gel electrophoresis view of the amplified gene, then the percentage and abundance of the different organisms isolated are indicated here, with Acinetobacter having the up to 10 number representing 63%, with the other species having different percentages. Then here we attempt to construct a phylogenetic tree to see how our sequence is clustered with the related sequence in the database. Discussion, isolation of the high number of diverse group of bacteria from the environmental sample in this work is not unexpected, as similar findings have been documented in a number of literature. And the ability of the isolated bacteria to utilize the waste transformer oil as carbon source will be attributed to their versatile metabolic ability of handling the complex organic structure, and accumulation of the PHA by the organisms in the culture containing waste transformer oil has further highlighted the viability of the concept of converting waste into wealth. In conclusion, the findings of this work has established the presence of diverse group of bacteria that are capable of utilizing waste transformer oil as carbon source to produce PHA, thereby providing the basis for exploring the potentials of waste transformer oil as cheaper alternative carbon feedstock for the production of bioplastic. With this, I say thank you to the organizers of this conference and the Ministry of Higher Education Malaysia and the management of University Science Malaysia for providing the grant. The research is actually ongoing, but we are able to present some aspect of it while we are looking forward to further with the other aspect of the research. Thank you very much for giving me.
Thank you, Shehu. Thank you so much for your presentation and all the best for your PhD, for your study.
Thank you very much.
Now, can you stop sharing? We'll move on to our Q&A sessions. So I would like to invite all the speakers to turn on your video.
Trying to stop the.
So we'll see you. All right. We have some questions here, apart from the questions being asked by the participants, and of course, being a chairperson, always take the opportunity to ask questions that I want to ask. But in the meantime, if you have any questions, please send it in to the chat box. I'll read your questions. Right, my first question to Prof. Futamata. Prof, are you okay? All right. Okay. No, interbacterial interaction is one of the very challenging topics, especially when you're looking at like the metabolic network. Well, it is really amazing what you have done. So out of curiosity, are you anticipating interspecies competition, which results in antagonistic effect of one species to the others, especially when you have like, let's say you have a new species that you would like to introduce into your SMC?
Say again, please. I didn't catch you.
Oh, sorry. My question is, yes. Are you expecting any interspecies competitions? Because you have shown that one metabolics reduce and then the other metabolics come out from the other species. Are you expecting any antagonistic effects? Like when one species is growing, it affects the other species of the bacteria. And what is the characteristics of the bacterial species that you are looking for to introduce into your SMC?
Okay, it is a very difficult problem. So we go to the phenomenon. So one bacteria replaces another strain's growth very, very strongly. And recently find this compound, interactive compound. And also the some cases is one bacteria has whole metabolic process gene. So under the batch condition, this bacteria can grow. But under the chemostat condition, this bacteria produce metabolite is negatively feedback. So in this case, is pure culture is broken. But the another strain coexist. It's like a mutualism situation can coexist each other. So now he's trying to analyze these relationships and then try to more stable situation using the another strains. But your point is very, very interesting and important point.
All right, because I think it's one of the challenging, especially when you have multi-bacteria in a system that you're trying to look for. And I think for environmental biotechnology, what the scientists are looking for is like to solve the problems in the world, the pollution and so on, especially phenols and so on. So I think that is one of the very challenging topics, like especially when you have a community of bacteria.
That's right.
Some are mutualistic exist, and some are just killing each other for the survival and for, you know, I think it's very interesting. I really have full respect for all the environmental microbiologists when this, because when we work in the medical field, we work on like apart from the gut microbiome, you have multiple.
That's right.
Yeah, other than that, you always work for like a pure culture where it's quite, but in a community, it's very challenging. All right.
Yes, I think so.
Yeah, thank you for your answers on that questions.
Thank you very much.
I have one question for Dr. Klima. I think your design of a jacketed system that you apply in the Antarctica is very interesting. I'm just wondering how do we apply this in the environments like in the future? So let's say the jacketed system is working, and we can create a huge jacketed system that you can apply in the Antarctica. Yeah, so what is your next plan and ideas?
Okay, next plan we have to try to make, actually the main problem is the source of the electric. So the next we try to make the motor from the wind to generate the electric. And then for the jacketed, yes, we need the jacketed machine. If not, everything will be freeze. We need to control because the temperature is around 10 to 12 degrees. But I think this machine can be used, successfully used during the summer. Only during the summer. In the winter, everything is freeze.
Okay, all right. So that would mean that we will not be able to have this bacteria and just spray it on the Antarctica and expect it to degrade the phenols and the diesels.
Because last time they do the experiment, they just spray the bacteria because the bacteria is local bacteria. But in the summer, there is a many snow, and then the degradation stop because of the low temperature. Because of that, we need the tank, not just spray the bacteria on the field.
Oh, okay. All right. Okay. So I thank you for your answers. And if anyone have any questions to Dr. Klima, you're more than welcome to unmute yourself and ask the questions. Or you can, if you are shy, you can always type the questions, and I'll help you to ask the questions. All right. Let's move on to questions to Dr. Rashan. All right. Actually, I have two questions here. One question is in the chat box, and you already answered that. And I think I would like to, if you're the question would be, how will the reductions in bacterial diversity in mangroves affect humans? And you have answered that. But I would like, if you're interested, for participants, if you're interested to look at the questions and answer from Dr. Rashan, you can look at the chat. And I would like to ask the other questions. Median sequencing might produce some errors, and how this error might affect the genetic diversity study.
Yeah, so in MinION, errors can come in different levels. One is in base calling. Base calling is that the principle of nanopore sequencing is change in that electric pulse when the sequencing reads are made. So if there are errors, probably they are going to assign into different groups. And therefore, I can see the abundance of one bacteria more than other due to sequencing. I think this has to be
Right. Out of curiosity, can I know how much is medium sequencing?
So actually, once you have the device, it depends on the chip. So each chip costs nearly 5,000 ringgits, and you can run 10 samples by barcoding, or more samples you can run by barcoding. And the good thing is you can wash the flow cell and use it at least twice. So we have actually used thrice. And also currently they will have some offers, like with this chip, you will get one more chip. So with that, it comes pretty okay, all right, to do sequencing using MinION.
Wow. Well, it's very interesting to see the technology because it's quite fascinating to look at it. All right. Thank you, Roshan. I'll move on to the next questions, which direct to Dr. Wan Iriani. Dr. Wan, are you with us? Yeah, okay. All right, so one of the questions from the participant is, what are the strategies that can be used to purify your silver nanoparticles, and how to determine its purity?
All right. So actually, we can use so many methods to purify the samples. Some people, they use column to get the pure compound that's majorly involved in the synthesis of a silver nanoparticle. But in our method, we very particular in terms of extraction itself, that we need to treat the sample very carefully at the beginning of the study. So we also compare with the positive control. There's a commercial silver nanoparticle. That's definitely pure, the pure silver nanoparticle. Then we can compare with our sample. Then based on the UV rays absorbent, and from the peak, actually we can indicate how pure our sample is as well. So there's some tips and strategy that I can share here.
When I look at your presentations using the polykit, it reminds me of teaching biodiversity to my students last time. They know that polykit can actually help in synthesizing silver nanoparticles. Do you have any plans to investigate the mechanism of actions, how the silver nitrate, if I'm not mistaken, that is being used to convert into silver nanoparticles? Any idea on the mechanism?
Yes, at this moment, my PhD student is working on the mechanism of action to look at in detail how the actual mechanism involved in the study. So he used a lot of gene expression, cell signaling approach to see how the silver nanoparticle can act as an antibacterial agent. So yes, because we are still running the project, so maybe you can share it in the future.
All right, great. Hope you present it in the next ICMBB. Well, I'm doing a promoting now for the next conference. All right. Thank you, Dr. Wan Iriani. I shall ask the next question to Ms. Rosina. What is Coprividis malaysiensis, and how do you obtain them? Because it's not clear how this organism is being used, was used to produce the compound, and in your methodology. So perhaps you can explain further.
Oh, okay. So it's a bacterial species, malaysiensis, which is found in the local environment in the Pulau Pinang Lake. They basically isolated it. My supervisor that I'm working under, So yeah.
Okay, all right. So is there any specific characteristics that you have with this bacteria?
Yeah, for the growth, it characteristics like it can produce the high pH or high pHA, and it is a very good the type of bacteria, it's wild type, but we have also the transformer type of this Copravides malaysiensis too. It has some differences about the bacteria.
So meaning that you can actually do some molecular.
Yeah, genetic modified bacteria.
Well, interesting. All right. So my next question is to Joel. All right. I'm curious about the rate of absorptions of the oil by the beads and degradations of the oil by the bacteria that you put in. So will the high absorbability of the beads overwhelm the bacteria for degradations? I'm just curious.
It shouldn't affect because if the bacteria is there, they should degrade whatever oil is that's on the beads or the fibers.
But you're expecting of like a closed system where you have the beads to absorb the oil and then the bacteria to degrade it, and then once it's done, and then you reuse it again, or rather than a continuous system.
Basically, sorry, I didn't understand what do you mean by continuous system. So basically, we're reusing the beads. Yeah.
The beads will be there, and the bacteria will continue. I'm looking at that open system where you have a degraded pollutions, oil going in, and the beads is absorbing, and then the bacteria continues to degrade. It's like open system, continuous system, rather than closed system where you finish one batch, then go to the next batch, then go to the next batch.
Okay, yeah. So yeah, I don't think it would affect too much, but over time, yes, the beads have a lifespan, I guess, of the absorbency. But because we only tested up to the 5th cycle, later on another student will be doing the more cycles, so that we sort of know how many times can this be reused.
Okay, that's interesting. All right, my last question to Shihu. For the isolated bacteria species that are able to produce PHA, any new species found in your study, or all the species are known for this ability based on previous literature? And which isolate would you suggest for the downstream works, and one which can degrade higher oil, or the one which can produce higher PHA?
Okay, thank you very much for that interesting questions. It's like a 3 questions combined together. The first one regarding whether there is any species that is new. To the best of our knowledge, there are 2 species among the 4 species organisms we isolated that we have never come across any literature. And these species are these organisms are the Proteus and the Serratia. We have never come across a literature, to the best of our knowledge, that reported the production of PHA. But as per the Bacillus and the Acinetobacter, we came across a number of literature, although very few, that reported the detection of PHA production, specifically medium chain length polyhydroxyalkanoids. And the second question is regarding which isolate will you suggest use for the downstream work? Actually, we look at the 2 major factors we study in our research: oil degradation in one hand and the PHA production. So we are trying to look for the compromise and organisms that has the ability to degrade the oil to a larger extent, and at the same time accumulating a reasonable amount of the PHA. Because when we look at from the one of the slide I presented, there are instances where you come across organisms that degrade high percent of the oil added, but producing very little PHA. So if we can have a compromising point where we can get one of the isolates, and I think that is what we are planning to do, to get an organism that produce as high and at the same time degrade as high oil as possible. Then lastly, the one which degrades, if I am still trying to get the last aspect of the question, one of which degrade I think I have addressed the last aspect of the question.
All right, thank you, Shihu. All right, it seems that we have a very shy populations of participants. All right, I'd like to invite Prof. Futamata can you give some advice to our participants, like our PhD students? I think many of the participants who will join today are working on the environmental biotechnology. Do you have any advice for them on what to go about, like what to look for the future directions in like bioremediations?
Okay.
Yeah.
Okay. Thank you, Chairman. So, as you know, the microorganisms is very, very diverse, and these microorganisms has a relationship each other. So we have to analyze each bacteria from the environment and try to understand how live in this site. And also we have to understand the relationships. So as you know, we analyze in the batch or pure culture system. The result from this pure culture system, this information is not so useful to practical situations.
Yeah.
We have to always thinking in natural system. This is the next step.
Yeah, I really agree on that, especially when you have like, a consortium of bacteria.
That's right.
Yeah. So I think that is one of the challenging topic for many of us, where we study like pure culture. Every time we identify the cultures, but when you apply that into the environment, it just doesn't work. So just thinking like, I think the community. But one of the challenges is like when you have a community of bacteria, and then when you try to analyze what is the like overall effect that it can help in degradations or bioremediations, that will be also another challenging, because you might not know where does this effect come from, whether it's from this bacteria or from the other bacteria, or it's actually a work together of a few bacteria in achieving the same effect. So that is very challenging. And do you have any suggestions to the students on?
So basically, please enjoy your science. It's very important.
I enjoy the science is one thing, but we also facing challenge in like getting funding, especially for the PI. For getting the funding from governments and from any institution is more and more challenging, especially during this pandemic. And the students are not allowed to go back to the university to do their research.
I think so.
Funding problems, chemical suppliers problems.
That's right.
But
yes, I think so. So through like this opportunity, so we had better collaborate with each other.
Yeah.
International collaboration is maybe very important to get the great fund.
Yeah.
And then grown up the student.
Yeah. So Prof. Futamata already sent out invitations to everyone who want to collaborate with you.
Of course.
All right. So I would like to thank our speakers and for presenting today. Before we end the sessions for today, I would like to invite everyone to turn on your video for us to take a group photos, please.
Of course.
All right, so let's turn on your video. And we have 3 pages here. Give your biggest smile. All right. Okay, so I don't know which page are you in, so we have to keep smiling until it's done. Ready? Smile. 1, 2, 3. Okay, I'm going to the next page. 1, 2, 3. And then 1, 2, 3. All right, thank you so much, everyone. Thank you for joining the environmental biotechnology and molecular biology sessions today. Please join us again tomorrow morning for our last day of the conference. And for MSMBB members who have registered for AGM today, please don't forget to join the AGM at 2 o'clock today. You should be receiving private invitations in the email. All right, back to you, Bunchi.
Thank you, Dr. Chia. Ladies and gentlemen, so it is now come to the end of the second days of the conference. So I hope you enjoyed the conference and found the sessions informative. So we hope to see you tomorrow in the morning, 9 o'clock. So for the MSMBB members who have registered to attend the EGMs later, please join us at 2 p.m. using the link that we have sent to you. So thank you everyone and stay safe.
Thank you.
Thank you.
Bye.
Bye.
Thank you. Bye.
Thank you.